Sheila ma'am, welcome ma'am.
Good morning. Can you hear me?
Yes ma'am, very good morning ma'am.
Very good morning to all.
Ma'am, can we wait for a few minutes ma'am? Yes ma'am.
No problem.
Because Dean ma'am will join and HOD from Zoology Manimegalai ma'am will also join, and there are only few participants ma'am. We'll wait for 10 minutes so that let the other
problem.
Also
You
join.
can take your time. Just inform me. I'm just in this meet only. No problem.
Sure ma'am. Thank you ma'am.
Thank you, ma.
Good morning, Dr. Sheila, ma'am.
Good morning.
Good morning, ma'am. Ma'am, we will start the session soon, ma'am. We are waiting for Dean, ma'am, to join, ma'am.
No problem. I'll wait.
Yes, ma'am. Thank you, ma'am.
Okay.
Sir, Gayatri? Good morning. We can start, Yamuna.
Ma'am,
Dean ma'am join பண்றாங்க ma'am.
Dean ma'am-ஆ?
Dean ma'am ஒரு 10 நிமிஷத்துல join பண்றாங்க ma'am. சரி. ஃபோன் பண்ணி சொல்லிருக்கேங்க ma'am. 10 நிமிஷத்துல ஜாயின் பண்றாங்களாமா? எஸ் மேம். நான் சொல்லி 5 நிமிஷம் ஆச்சுங்க மேம். நான் திரும்ப வேணா ட்ரை பண்ணி பார்க்கறேங்க மேம். வரட்டும். நோ ப்ராப்ளம். ஓகேங்க மேம். வந்திருக்காங்க ஒரு 20. இந்த வெஸ்ட் பெங்கால்ல வந்து மேம் டீன் மேம் ஜாயின் பண்ணிட்டாங்க. மார்னிங் மேம்.
Good morning. Good morning.
Good morning.
Good morning to everybody. Good morning, pa.
Okay.
ஸ்டார்ட் பண்ணிக்கலாமாங்க மேம்?
டீன் மேம் கிட்ட கேட்டுட்டு பா.
எஸ் மேம். மேம் ஸ்டார்ட் பண்ணிக்கலாமாங்க மேம்?
Yes, pa, we can start.
எஸ் மேம், எஸ் மேம். யமுனா?
Yes, ma'am. Yes, ma'am. We'll
start
start.
Check. Respected Dean, School of Biosciences, heads of the organizing departments, distinguished guests, organizing secretaries, faculty, research scholars, and all the participants. Warm morning. I am Ms. Angeline Priscilla of MSc Biotechnology. Happy to welcome you all to the final day of the UGC Stride-sponsored 6-day training program on environmental sustainability and research ethics. Before we begin today's training sessions, let us invoke the Lord Almighty's blessings on all of us through a prayer song. I invite Ms. Carmugel Madimurial of 2nd PG Biotechnology to grace this occasion with a prayer song.
கல்லார்க்கும் கற்றவர்க்கும் கலிப்பருளும் கலிப்பே காணார்க்கும் கண்டவர்க்கும் கண்ணளிக்கும் கண்ணே வல்லார்க்கும் மாட்டார்க்கும் வரமளிக்கும் வரமே மதியார்க்கும் மதிப்பவர்க்கும் மதி கொடுக்கும் மதியே நல்லார்க்கும் பொல்லார்க்கும் நடு நின்ற நடுவே நரர்களுக்கும் சூரர்களுக்கும் நலம் கொடுக்கும் நலமே எல்லார்க்கும் பொதுவில்லிடம் இடுகின்ற சிவமே என் அரசே யான் புகழும் இசையுமனிந்தருளே இசையுமனிந்தருளே இசையுமனிந்தருளே.
Thank you, Ms. Carmugel. At this time, I would like to call Ms. Dhanashree of MSc Biotechnology to introduce the first speaker of today's morning session, Dr. C. Sheila Sasikumar.
It's my great privilege to introduce the eminent speaker of today's first session, Dr. C. Sheila Sasikumar, Director, Clinical Research, SS Healthcare, Chennai. Dr. Sheila Sasikumar has more than 3 decades of experience in both academics and research. Her area of research expertise includes clinical research, biochemistry, nanotechnology, 3D bioprinting, molecular biology, and natural products bioactivity. She has published about 62 research papers in reputed international and national journals and a patent on diabetic wound dressing material. She had published a significant number of book chapters and had presented her research work in various national and international conferences. Dr. Sheila Sasikumar has received Professor E.P. Odom Gold Medal in 2007 for commendable research activities and has won the Distinguished Adjunct Faculty, Department of Biochemistry, Savitha Dental College and Hospital, Chennai. Dr. Sheila Sasikumar is one among the most significant personality in the field of clinical research and had successfully completed many clinical trials, among which the South Korea-based clinical trial study on the effects of diabetic foot ulcer improvement using the 3D bioprinting technology, a comparative clinical study to evaluate skin and soft tissue infections using a novel, non-invasive, fluorescence-based imaging device, and the effect of oral protein supplement PRO 360 on diabetic foot ulcer patients are noteworthy. She has effectively completed a substantial number of research projects that mainly focused on diabetic foot ulcer and its management using herbal formulations. We welcome you, ma'am.
Thank you for the warm introduction, Ma. Let me share my screen and then I'll speak. Is my screen visible?
Yes, ma'am, it is visible.
Is it moving?
Yes, ma'am.
Right.
Perfect.
Perfect. Thank you, Ma. Hi, good morning to one and all who have joined in this virtual webinar meet on environmental sustainability and research ethics conducted by Avinashilingam University. Respected Chief Patron, Chancellor, Vice Chancellor, Registrar, Project Coordinator, advisors, conveners, organizing secretaries, and my dear participants. I feel deeply honored today, as well as very privileged, to welcome you all for this webinar meet on air pollution, climate change, and human health, a very hot topic indeed, which we need to understand as an individual, and what are our responsibilities and how to overcome this as an individual person. As a resource person, for series of webinars conducted by hospitals, universities, colleges, industries, etc., I shared my experiences regarding health, research and development, bioprinting, especially on diabetes, diabetic foot ulcer, etc. This is a first opportunity, and I was waiting for such an opportunity to talk about climate change and health, which every common man should understand, and it is indeed a need of the hour. So let us go to the topic: air pollution, climate change, and human health. The student has given a very warm introduction. I have just projected the links about me, where you can come across my publications, mostly on the recent advancement in the health technology. I'm a person working on a clinical research, especially on the digital healthcare sector, or you call it as a digital healthcare products, that is concerned in the recent innovations, especially in the post-COVID era, and this era is called the technology era. So usually I start with this. Women empowerment is one of the strategic challenges for sustainable development. So if a woman gets educated, it's the entire society gets educated. Why am I speaking this about this today is, if women knows about the climate change and what happens to the health and how we should protect our environment, I think the community itself will understand. That is the main aim here. So I'm here today as a women's service entrepreneur, and the link I have provided here is the link where I have shared my experience about the role of women in research in a talk on women in research in AICTE program conducted by SRM University, where I have provided the opportunities for women in research. So my organization, my consultancy, SSL Care, is connected with clinical research and education. Education is a passion. Having 3 decades of experience and research, this helped me to come as a women's service entrepreneur today. My mantra in clinical research is translation of products. My main aim is converting the products or translating products from bench to bedside, that is basic research to the commercialization. And also during this process, I see to the quality, efficacy of the product, patient safety, ethical and data integrity is what my business demands. Our services are mainly on clinical trials of pharmaceutical, herbaceutical, cosmetic, AI, machine learning products, and software, apps, etc. in healthcare sectors. Also development of the products. We help the researchers to start from the idea to the venture, from the idea to the complete products. And also we do educational services, and we have started mentoring programs along with the foreign university, especially with the Astella Pharma, regarding the opportunities that are available for our students to pursue the higher education as well as the job opportunity. And we do collaborative research where we enter into an MOU and help them to convert the products from bench to bedside. So these are the things about my organization and what I do. And coming to our talk on climate change and human health, if you see the screen, I have projected what is the reason for the climate change and what happens and how our health is affected. This one picture will depict what is the impact of a climate change, even a small fraction, or I can say even a small degree of climate change that is happening has a worsening impact on our human health. Not only the human health, the entire ecosystem gets affected. So that is what we are going to see today. I'm not going to talk about the process, etc. My concern is you have to understand the effects on the human health, which is our main concern here. So what is this climate change? Climate change, I can say this is the biggest challenge that is facing the humanity, and not only a biggest challenge, it is the single biggest challenge we can say that is facing the humanity. And all the clinicians, surgeons, and all the healthcare group are working for this, how we can prevent it and what are the things that are happening, and researches are going on in this area. The IPCC, the Intergovernmental Panel on Climate Change, has concluded that to avoid this catastrophic health impacts, so how can we prevent this system changes? Because it's not that one particular phase is going to get affected. It's an ecosystem. Our planet is a complete ecosystem. If there is an impact in one factor, it magnifies and it has its impact all over. So the entire system gets disturbed. So to prevent millions of climate change related health, the world must limit the temperature rise. So it is in our hands, because this happens both natural as man-made. So man-made can be limited in order to prevent a rise to 1.5 degrees C. So past emissions, already it has happened. So past emissions have already made a certain level of global temperature rise, and other changes to the climate inevitable. At least from today, we can think of how to prevent this. So what is about this climate change? So see here a picture, the earth here is afraid. So it is thinking, oh, when the temperature rises, what happens? So what is this climate change? According to UNFCCC, that is United Nations Framework Convention on Climate Change, who are working on this, it is defined as a change of climate which is attributable directly or indirectly to the human activity that alters the composition of global atmosphere. So the global composition is affected, the atmospheric composition gets affected, which is in an addition to the natural climate variability observed over a comparable time period. So simply we can define it, any change in the temperature due to the emission of greenhouse gases can reflect or can have an impact on the change in the temperature. So which is called climate change as a simple term. So what are the major impacts? Why are we worried about this? So when there is a climate change, or when I say there is a rise in the temperature, what happens is that there is an impact all over. So this one picture, you can understand what are the systems that are going to get affected. So what happens when there is a increase, there is a decrease in snow cover, change in the precipitation pattern, which is very important when we are talking about the air pollution, asthma, respiratory diseases, chronic pulmonary diseases, etc. Increased evaporation rate, more droughts and heat waves, forest fire, so deforestation happens naturally, bleaching of coral reef, so what happens? The marine ecosystem gets affected, increase in the ocean temperature. So we are going to lose many of the living organisms. So again, the aquatic ecosystem gets affected. So these are the main global impact on the climate change. Whenever there is a climate change, this happens. Melting of ice and increase in the sea level, or rise in the sea level, which we can say. So in the year 2016 to 2020 is considered as a hot season in the world history. So this was reported by New York, Geneva. Climate change has not stopped for COVID-19. So 2016 to 2020 is said to be the warmest 5-year period on record. So we have come across, and now we are in 22. So we know COVID came in picture. The first COVID, the second COVID, we had series of pandemic things happening. So in the first COVID, second COVID, etc., we had all lockdowns, and we were completely from chalk and board. We came to the digital transformation. We are into the digital things everywhere. Now I am sitting here in Chennai, you are in Coimbatore, we are discussing about this climate change. All happened during this lockdown period. So lockdown related fall in emission will not reduce. We were thinking that, but small things has happened that has to be accepted. During this lockdown, there were some changes that were happening in the nature, because the emissions, especially the automobile emissions, all came down because we were in lockdown period. But what they say in research is lockdown related fall in emission will not reduce carbon dioxide concentration. That has happened already. The impact is already there. So all these things which had happened will not change, but we can do some changes at least in the further future conditions. So greenhouse gas concentration in the atmosphere are at record level and continue to increase. We are using AC, air condition and other systems where already CFCs are there in the picture, which is going to have its impact in the atmosphere. So emissions are heading in the direction of pre-pandemic level following a temporary decline. Remember, there was only a temporary decline during this COVID-19 lockdown period, but there was also a decrease in the economy because we were under lockdown, and work was not going on. The profession was all professions, we had a stop there, but there was a small impact on the nature. The world is set to see its warmest 5-year record in trend, which is going to continue and not on track to meet agreed targets. So the Paris Agreement, which we'll see, they made an agreement that definitely all countries should come in contact and do some remedies, and global measures have to be taken to prevent this rise. So what are the fast facts? I said I'm going to talk about only the climate and the health impact. So what are the fast facts? The World Health Organization, please see the link that I have provided here. The World Health Organization has already jotted down what are the things that has happened due to this climate change. Number one, every year, the environmental factors take the lives of around 13 million people globally. So we were thinking that death is happening due to cholera, due to tuberculosis, or due to COVID, or whatever it is. No, there are, in different parts of the world, death is happening because of climate change and other environmental factors. So I told earlier the Paris Agreement, if the Paris Agreement objectives are met by the world leaders, then we can save about millions of lives in a year. And also we can reduce the air pollution alone by the year 2050, so that we can give a healthy planet to our future generation. And the next fast fast-tracked impact by who, what they say is the value of health gain from reducing the carbon dioxide emission alone would be approximately double the global cost of implementing the mitigation measures. So we are going to spend a lot to reduce all these because already we have done the emission. So the cost is going to be very high. And 90% of the people all around the world are breathing poor quality of air that is caused by the air pollution. And transportation produces about 20% of global carbon emissions. So I think nowadays we are going to switch over to other type of fuels, and electric vehicles are going to come in picture. So we have to control our transportation, which we'll see under the remedies. So transportation produces 20% of the carbon emissions. Then the most sustainable production, that means system to produce, package, distribution of food, generate third of greenhouse gas emission. So we need to think about more sustainable production that would mitigate the climate impacts and have some nutrition content in our food that is being produced. So because of this, 11 million premature death is happening. Health systems are the main line of defense for the population faced with emerging health threats, including from climate change. So we must build a good health system, and it should not get affected due to the climate change. So less than 2% of multilateral climate finance goes to health projects. This is one area where everyone should have a concern. There are so many projects being signed for this climate change, air pollution, and its impact, and especially on the respiratory diseases, etc. But whatever it is, only 2%, less than 2% of this goes for the exact work. So we need funds to work on this and prevent all these changes. Healthy societies rely on well-functioning ecosystem to provide clean air. Clean air. Remember in Delhi during this COVID time, we know how it happened. And the second COVID time, we were searching for the oxygen cylinders, and they paid double the cost or triple the cost for the oxygen. Remember that very well. So we need a clean air, fresh water, medicines, and food security. Clean air and fresh water is the right of every human being. So we don't have the right to pollute air, which is a very essential component for all the living system that is there in the world. So these help to limit the disease and stabilize the climate. But biodiversity loss, I think zoology professors, botany professors are all here. Biodiversity loss is happening at unprecedented rate, impacting human health worldwide and increasing the risk of emerging infectious diseases. The main goal of my talk is because of climate change, vector changes, vectors quality, that is habitat, everything changes, leading to many infectious diseases in mankind as well as in the animals. So these are some important facts that World Health Organization has commented, and we need to understand and take an important measures. So what are these health impacts categorized as? It is categorized as direct factors or indirect factors. When there is an impact on the health due to climate change, directly it affects how thermal stress. Thermal is increasing in temperature, causes mental stress, physical stress, etc. Then it may cause death or injury in floods and storms are the direct implication of climate change. Whereas indirect is the main concern here. Indirect, what happens? It causes vector-borne diseases. This is very important part. Vector-borne diseases, waterborne pathogens, water and air quality, availability of food, and the quality of life is affected. These are the indirect impact on the health system due to the climate change and air pollution. So climate change is already affecting vector-borne diseases transmission. See, this is a picture. It's a very nice picture. You should understand. As and when the temperature increases, we were thinking that only rainy season we will have the disturbance of this mosquitoes, but it is not like that. As the temperature increases, this vector, because plasmodium or this mosquito is going to be a vector which is going to carry the protozoa, which is the exact thing. So this has some changes in its body, and it gets adapted to this hot climate, and as a result what happens, it starts to create infection. So climate change is already affecting vector-borne disease transmission, and its widespread and impact are likely to worsen. So in the ongoing phase of this climate change, we must definitely intensify the efforts to prevent and control the vector-borne diseases. So this was a recent paper that they found this. It's a research paper. You can see in the Nature. Climate change and the challenge of vector-borne diseases prevention and control. So vector-borne diseases, as I told you, direct climate change, if you see, whenever there is an increase in temperature, the vectors changes its distribution boundaries. That means, of late I came across mosquitoes during the Second World War, especially when the people were staying in the tunnels. I came across this. The mosquitoes were able to adapt into the tunnel area because the population were living in that because of this World War time, and they were able to adapt themselves in that atmospheric condition. As we all know, when we go to the tunnel at higher altitude as a lower level, the atmospheric concentration varies. These mosquitoes were able to adapt themselves in the tunnel, and all type of vectors started to establish or invade in that area. So this explains you they can have the ability to adapt in any type of environmental conditions. So whenever there is a temperature increase, they change the boundaries. That means from a higher altitudes and latitudes, they cross the boundaries. So a mosquito by somehow it can cross from the region of Africa to the another country. So this happens, and it causes an effects on the biology and the physiology process. Or else, because of the increase in temperature, what happens? It brings about an accelerated process of pathogen development. There is an increase in the replication process. As a result, what happens? N number of pathogens are produced within a small period of time, and completion of cycle happens at higher altitude as well as higher latitudes. So this shows whenever there is an increase in temperature, they don't have any problem. They start to adapt and they start to spread their infection. That is one thing you have to keep it in mind. Then when there is a global wind pattern alterations, so change migration of certain vectors. So when the wind is going to have its impact. This is also one of the environmental risk factor. What happens? It starts to migrate from one region to another. Then global precipitation pattern changes in the length of the season that vectors can survive. So vectors are able to survive and they start to spread the infection. Changes in the humidity effect on vector lifespan. Lifespan of the vector increases. If the lifespan increases, the ability to give more infection is going to be increased. So there will be a change in the genetic composition of vector population. So if you see any type of factor that is seen due to a climate change, one factor called climate change, or I can say a small degree rise in temperature can have its impact on variety of factors. So changes in the transmission, behavior, geographical distribution, and incidence of vector-borne diseases can cause mortality and morbidity in most tropical countries. So please remember, India is also a tropical country here. Indirect climate changes on disease vectors. Now, whatever I've shown you is a direct effect. Now, indirect effect, it can be when there is a drought. What happens? Reduction in the density of water-related vectors. When drinking water becomes a scarce, what happens? More chance of guinea worm transmission and changes in the distribution of rodent reservoirs, sandfly, thus affecting or causing black fever. Or other changes in the vegetation because of climate changes affects text flight distribution and risk factors. So what happens? It causes bacterial diseases, vector which is called viral tick-borne diseases. When there is hydrological changes, formation of more brackish water, we know very well. As a result, what happens? Breeding of lacunae, what you call the fresh breeding of brackish water species, all these things, and changes in the riverbeds affect the ecology pattern. And whenever there is a changes in the agricultural practice, this is also one concern we have to see, or changes in the cropping pattern or too much of chemical pesticide application, etc. What happens? It has its effect on mosquito-borne diseases and snail-borne diseases. So if you see, there is a direct impact as well as indirect impact on vector-borne diseases. So how climate affects vector-borne diseases? We have seen transmission, geographical pattern, and multiple pathways, including its direct and indirect effects, not only humans as well as in animals. So climate change can alter the entire ecosystem, habitat, including urban habitat in which vectors or non-human hosts may thrive or fail. So arthropods. So we know arthropods, especially the cockroaches, etc., and other vectors called ectotherms. It is expected that vector abundance, survival, and feeding activity will increase with the increasing temperature. As a result, the development of pathogen and the transmission season will also increase. So see, if you go on analyzing the system, you can see the magnification happening from the lower level to the higher level of animals. It doesn't stop at one particular point. It is a chain of events. I can say it is also an example for biomagnification that is happening here. So increased temperature. Now you may ask me, why are you concentrating on mosquito alone? Dear participants, even today, malaria is prevalent, dengue is prevalent. Recently, we saw during the third COVID time, since I was working in the COVID hospital, we know that along with the COVID cases, we saw n number of dengue cases also coming in picture. And in some cases, both were happening. So there should be some factor that is going to accelerate all these processes. So increased temperature due to climate change. Now, if you see mosquito survival, I told you the vector has its ability to adapt. So what happens is that its biting frequency may be amplified in certain higher temperature environment. So biting frequency in the sense, zoology persons are here, we all know mosquito put forth its proboscis and it sucks the blood. Now, we do not know, we can't see or we can't tell how much time the mosquito comes, each mosquito comes and bites us. But researchers in the worldwide had proved that because of temperature increase, these mosquitoes have the ability to take more of nutrients for them. So what happens? The biting frequency, the number of times the mosquito bites a human or a host increases because of the increased temperature. And also, as I told you earlier, increased temperature becomes one of the factor to increase the replication rate in these malaria causing parasites. When there is a change in the precipitation, what happens? You can see rainfall happening and there is pool of water stagnant. In the stagnating water, what happens? The mosquito starts to breed. So extreme rainfall events can destroy the infrastructure and place at higher risk for. So when there is a flood or when there is an extreme rainfall conditions, now the people who are living in the lower areas are also affected and they are exposed to more number of biting frequency of this mosquito. So this is one of the important example how climate change can affect our health. So not only malaria, so we see that climate change is expected to cause additional 60,000 deaths per year due to malaria. Not only malaria, even dengue. The dengue that is caused, the vector is going to be the Aedes mosquito. The rates of dengue transmission may actually increase in the region that are more prone to drought. So when they are more prone to drought, what happens? They starts to breed in as such, these mosquitoes breed in the clean water and it starts to breed in the drinking water and other things, household water storage containers, etc. Now, if you see the picture here, climate change happens, the impact happens due to two reasons. One, poverty. So poverty is also one of the factor where the health impacts is going to be directly influenced. Another one is diseases. So when there is a climate change, poverty, what happens? They don't find a place. They don't find a place to live. Financial insecurity, all these things happens, especially in case of people living in the low income areas. In case of diseases, all these factors which I showed you can cause. As a result, what happens? It is a cycle. It is going to be a cycle here. Climate change affecting the population either two ways. In both the ways, it is going to reflect only on the health and disease. So climate change exacerbated diseases by providing a very warm condition for the vector to survive. Even fungal infection is common at a warmer climate. Then poverty by exploiting the resources and causing monetary difficulties in country. Then lower financial stability and limited resources storage. As a result, also this happens. So what are the risks we are going to talk about and who are at risk most commonly? We know now we have a picture that climate change is going to affect in myriad of ways, leading to death or any non-communicable diseases or even communicable diseases, whatever. So who are at risk? Mainly children, women, ethnic minorities, poor communities, migrants, displaced persons, and older population, and those who are already affected with some underlying conditions, for example, cancer, arthritis, or any disorders already who are under treatment. So this is also a picture from CDC, Center for Disease Control in the global part. Now, if you see, I told you who are vulnerable, who are more affected. Now, if you see vulnerability, we can think about the factors, the pathways, and the health system. So climate change impacts health both directly and indirectly. Now, if you see the vulnerable factors, demographic factors, demography is what I told you, gender, age, I told you children, older age, women, etc., pregnant women, all those people, geographical factors, tropical climate maybe, so based on the latitude and altitude and all those conditions, then biological factors and health status. So here is what we need to understand about the role of immunity. If you are if you are well-fed person, that is not overfed or underfed, a well-fed person having a balanced diet of sufficient amount of macronutrients and micronutrients in your diet, you have immunity. So the biological factors, which is called the health status, the physical and mental being. If you are in that category, no problem. But if there is an alteration, for example, children suffering from protein energy malnutrition like kwashiorkor and marasmus, so they don't have sufficient proteins in the body for building up, and as a result, the immunity is very less. In those cases, it goes to be a problem. Socio-political conditions, socio-economic factors. So generally, high income people, they have the ability to use they use the monetary resources, whereas low income people, they have limited availability of resources and poor availability of hospital facilities, etc. So exposure pathways, when I'm going to talk about, is stress, thermal stress, air quality, water quality, food security, vector distribution, etc. That comes under the exposure pathways. Health system, which has to govern all these factors. So these two factors have a direct role on health risk, producing injury leading to morbidity and mortality, heat-related illness, respiration problem, waterborne pathogen diseases, zoonosis, vector-borne diseases, malnutrition, NCDs, which is called the non-communicable diseases, and mental and psychosocial health. Here also research is going on. Thermal stress has a direct impact on the mental well-being of individual. So please remember, now we are in the summer season. We need to take utmost care about our health. So climate change and health way, this is from European Protective Agency regarding the climate change. So if you see the same thing, but here I want to tell you one thing more also, is cardiopulmonary illness. So not that only respiratory system gets affected, along with the pulmonary system, it is cardiac also gets affected. So remember this, and this is about the whatever I told you about the age and gender, race and ethnicity, poverty, housing and infrastructure, education. So today we are touching this education part. I can say this is also a educative campaign to understand and create an awareness on the climate change, access to health care, etc. So all these things can have its impact on the human health. Remember, we are sitting inside the AC room, or we are sitting inside a classroom, or we are sitting inside a IT industry. We do not know what is happening outside. You should also understand nature. What is nature, how it gets affected. Only you will know the reality of the picture. Now, this is one part where I'm doing a few research work in this, because I want all people to understand, especially young children. They should understand and give some ideas to help this impact on the health changes that is happening due to climate alteration. Now, this is also given by the European Protective Agency for Climate. Now, if you see extreme heat, flooding, tick-borne diseases. I'm going to concentrate here only on the tick-borne diseases. So we know when there is an extreme heat, all this going to happen. So tick-borne diseases are also happening simultaneously. Disease carrying ticks earlier in the season and in new parts of the country, especially called Lyme disease. They have found out that it is happening due to this climate change. Not only vector-borne diseases, tick-borne disease, especially called Lyme diseases. Also, whenever there is a climate impact, flooding causes gastrointestinal illness and injuries. So there can be worm infection, or what you call tapeworm infection, or malabsorption, and all these things happening here. Sometimes bloody stools will also be released due to some waterborne pathogens entering in the body. So this is also another thing we have to understand, that not only vector-borne diseases happens, it's also a tick-borne diseases that is going to happen due to the climate impacts, exposure. When there is a change, we have been exposed. Definitely, you cannot run away from the atmosphere. You need to survive. You need to breathe. Whatever the quality of air, you need to breathe. So when you are exposed, what are the outcomes? That is what we are seeing today. Now this picture, please see this picture very carefully. Rising temperature, more extreme weather, rise in the sea levels, increase in carbon dioxide. So all these environmental factors causes the first level of changes. See, air pollution, vector ecology, which we saw today, increasing allergens, water quality impact, water and food supply, degradation of environment, extreme heat, severe weather. Each one causes one type of diseases or the other. So change in vector biology, ecology, I told you malaria is the most concerned, followed by dengue. Apart from this, you have encephalitis, hantavirus, Rift Valley fever. I told about Lyme disease, which is a tick-borne disease, chikungunya, and West Nile virus. When there is an increase in allergen, we all know asthma will be the common cause, common disease. Now, when you see here, water impacts, when it's waterborne pathogens or poor quality of water, we know cholera will be the most important one, followed by cryptosporidiosis, campylobacter, leptospirosis, and algal blooms. And when there is a shortage of food supply, protein, energy, malnutrition, or any, in case of adults, it is going to be malnutrition, devoid of proteins, you cannot build the immunity, and diarrhea. When there is a degradation, forced migration, yes, if that environment is not suitable for me to live, for the human to live, what we do, we start to migrate from one area to another in order for our survival. So we know Darwin's theory is survival to the fittest. We need to change ourselves. So civil conflict, mental health impacts. So heat related, when there is an extreme heat, cardiovascular failure. When there is a weather changes, injuries, fatalities, and mental health impacts. Air pollution, COPD, and cardiovascular diseases. So this one picture, I think we can understand if any one of the factor in the environment is altered, see what are the impacts that is going to create. Unknowingly, we are here and we never bother about this environment. It is so sad that we should take at least today some measures. So what are the determinants of vulnerability? I said vulnerable population, but there should be some factor called vulnerability. எவ்வளோ நேரம் நான் expose ஆயிருக்கேன், how much I am exposed to this condition, and once I exposed, am I going to become sensitive or am I going to become adaptable? This is what helps us to withstand and survive. So exposure is what you call your when a person comes in contact with one particular factor, it can be psychosocial, chemical, biological, or physical factor, any stress related factor, etc. Sensitivity is how well I get accepted or not accepted. So if I'm affected and I'm not able to withstand, that is going to be the sensitivity factor. Adversely affected or beneficial. Okay. Ability to adapt. Okay, my system gets adapted. I can survive in this type of change. So adaptive capacity is the ability of communities or people to adjust to such a potential hazard that is happening in the environment and able to respond to the consequences. Okay, there is a climate change happening. Okay, there is a waterborne diseases. How well I get adapted and creating an immunity is called the adaptability. So vulnerability of human health to climate change, health outcomes. So this is called determinants of vulnerability: exposure, sensitivity, and ability to adapt. So exposure, if you see, low income population, occupational groups, and people in certain locations are exposed. Sensitivity, what happens in case of pregnant women, people with pre-existing medical condition, and children are more sensitive. Ability to adapt, how well we are able to adapt. Older adults have very limited capacity to adapt the extreme weather changes, and people with disabilities. So disabilities, we must give consideration to them because they may not able to adapt. Then indigenous people who rely on the substance of food have limited options. They cannot come out because their profession or their environment is like this. So these are the determinants of vulnerability. There this is about the changes and in its impact on the human health. Also, I see that food safety and nutrition also gets altered whenever there is a climate change. So extreme weather events can disrupt or slow down the distribution of food, and higher air temperatures can increase infections caused by salmonella as well as other bacteria, which leads to food poisoning and causing gastrointestinal problems, and in severe cases it causes death. So this is one of the change that is happening due to the higher concentration of carbon dioxide that is prevalent in the atmosphere. So another example I can give you on food safety, whenever there is, I told you, was salmonella increases. Also, whenever there is an increase in the this temperatures, already we have chemical contaminants prevalent in the soil, that is in what you call land pollution, water pollution, air pollution. Now, these contaminants can be heavy metals, especially mercury and lead. And in case of aquatic system, what happens is that higher sea surface temperatures, that is, whenever there is an increase in temperature, the surface will be very hot. And as you go down, there is a gradation in the temperature. So the surface temperature, higher the surface temperature will lead to higher mercury concentration in seafood, and extreme weather events will introduce contaminants into the food chain. Now, if you're going to take fishes, consume the fishes or whatever the non-vegetarian food from those sources, what happens? The mercury, slight concentration of mercury enters into the aquatic ecosystem, and ultimately we are going to enter with an increase in the mercury concentration. So this is one of the change happening in the aquatic system, and salmonella infection is another one. Then another one that is happening is because of the constant use of chemical fertilizers to increase the yield, to prevent the pest, rodents, etc. It causes it causes an increase in the carbon dioxide in the atmosphere. And then higher concentration of this carbon dioxide that is present in the atmosphere can act as a fertilizer. How it acts as a fertilizer is that it can be taken up by the plants for its carbon cycle, etc. We have studied about the C4 pathway and C3 pathway, etc. But when too much of carbon composition increases, the protein composition decreases, or the essential minerals that is required, phosphorus, potassium, calcium, etc., decreases. As a result, the crops which are surviving, for example, rice or potatoes or whatever may be, will not have sufficient nutrients as it is expected in that vegetable. So it becomes less nutritious. And when you are going to get a less nutritious food, of course, our nutrient content also decreases. So extreme events such as flooding and drought created challenges for food distribution if there is a disturbance in the transportation process also. So if you see the farm to table, now this is a picture which you are seeing now is from farm to table, how the changes affects from the production to consumption. So here, if you see, the production gets affected, increase in the carbon dioxide content. Because of this, the nutrition content decreases, and there is a disturbances in the transportation process. Because of less nutrition content, there is a greater food spoilage, etc. So everywhere there is an alteration because of one factor change. Now, the next part we are going to see is the air pollution. Now, who estimates 7 million people die annually? The World Health Organization has projected that 7 million people are dying annually because of air pollution, making it as an important risk factor for premature mortality. And also it is estimated 4.2 million death of pediatric population around the world because of this air pollution. So you can see this, here is the link I have provided here. So why air pollution is a main concern? Changing climate changes the weather pattern. As a result, what happens? Outdoor air pollutants also increases, and particulate matter, which is called as PM, also increases. Then increase in CO2 we saw now. Because of the increase in CO2, there is a less amount of protein in the plant, and also it releases the aeroallergens. Allergens, that is, they are along with the pollen. Pollen, of course, is present. Along with that, there will be other allergens that are released by these plants, which becomes one of the asthmatic factor. Then changes in the outdoor quality can have its impact on the changes in the indoor quality, because both the pollutants and the allergens can enter into the enter or infiltrate in your home, schools, and buildings, and this causes some allergy diseases in children. So as a result, what happens? It limits the productivity either in the office or at school or at your workplace, whatever it is, because of asthma episode. So that is why I have shown here there are more children having this nebulizer. So that is one of the risk factor we have to think about. So in India, in our country, if you see, the major sources of air pollution can be natural sources or anthropogenic sources. So we know what are the natural sources. I'm not going to discuss here. So if we natural sources, of course, if you control anthropogenic sources, I'm a person, I believe that natural sources also starts to come under control if we limit man-made activity. So air pollution can cause short-term, medium, or long-term impacts on human health. So short-term impacts: irritation of the eyes, throat and nose, and respiratory infection like bronchitis, pneumonia, etc. Long-term air pollution, as I said, chronic pulmonary diseases, cardiac diseases, pulmonary cancer, and even damage to other essential organs like brain, liver, kidneys, or your nervous system. And more vulnerable persons should be children, elderly people, and pregnant women. So this is one picture. You can see the non-communicable diseases, underlying mechanism linking, how there is a link between a non-communicable disease and air pollution. So whenever there is air pollution, what happens? NCD, most important NCD is cardiovascular disease, respiratory disease, cancer, and diabetes. Now we stand second in the world in case of diabetes after China. So we have to control that. All leads to mortality, morbidity, and large sum of money is being thrown in the healthcare sector. And sometimes what happens, since I'm working in a pediatric center and diabetic foot ulcer hospital, if diabetes is not controlled, diabetic foot ulcer, and you may lose your toe, 4 feet, leading to amputation, and your life becomes dependent, which ultimately causes psychological problems, depression, stress, and all these things, because you are dependent on other people for all of your work. That becomes very sad. So we need to control diabetes. So loss of human capital, loss of physical capital, all these causes impact in the economic growth. So air pollution, this is one picture we need to understand. As a women, we should advise other women and other women population that during pregnancy, it will have a direct impact, causing oxidative stress, inflammation. And if you are a biochemist person, you can understand how it creates the stress and leading to impairment in the beta cell dysfunction, neurohormonal dysfunction, inflammation, stress, and imbalance of gut microbiome. Ultimately, all the factors, whatever I'm going to talk about, it is going to create a resistance in the insulin production, leading to gestational diabetes. So gestational diabetes is caused mostly by this air pollution. Please have a check on that. So what are the remedies? I hope I have taken time. Let me finish. This is a last part I'm talking about. Remedies. So till now we understood that what are the changes that is going to happen on different things, but ultimately it is going to affect our health, human population, and also our economy gets affected. So how do we control? What are the remedies that you can take about? Reducing emission of greenhouse gases will be the major thing in our concern through better transport, food, and energy use choices to improve the health. Particularly, we need to reduce the air pollution, increase the quality of air. Poor quality of air, we cannot survive. So what are the remedies you can talk about? Opting off renewable energy, promoting public transport. Still, we don't use that. Sustainable mobility. This is one of the things that World Health Organization is stressing upon. Increased number of journeys very nearby by walking, cycling, or decrease the number of flights and increase the train journey. So promoting ecological industries like agriculture, fishing, and livestock farming, responsible consumption, which lies in our hand, and 3R rules. I think everyone is familiar with this: reduce, reuse, and recycle. This 3Rs should be kept in mind even when we are using the throwing the garbage also. Please keep this in mind of removing not to use the plastics and use those things which are readily biodegradable. Green business. So if you see my PowerPoint, I have projected green color because my ultimate aim is the next generation should have a green earth in their hand. That means they should live happily, and the quality and the other things should be very well so that they can be happy. Happiness is dependent on that. So green business you can do: rooftop gardening, balcony gardening, community forestry, community or corporate tree plantation activities, gardening in the premises, land use planning. Now we do vertical gardening in case of offices and other things they are doing, pavement plantation, educative campaigns, etc. Then afforestation can be done to prevent the deforestation, diversifying crops so that they can adapt to the climate changes, investigating and developing innovative solutions to prevent all these damages, and action plans. Not only that, all the action plans that we jot out should be implemented, and remedies should be done as soon as possible because it's an emergency condition. And the last part is as I understand, because I said earlier, I got this opportunity to speak and I was waiting for such an opportunity to talk about, because I want to stress this. As I understand, the responsibility lies in each and every individual to make our planet evergreen. Our future generation should be happy. So small remedial measures can be done. And this is a small measure from my end, where you can see my terrace garden, which is my paradise, about 640 square feet, not very big, but still I can use that and create my own, produce my own vegetables for my family. I cannot produce massively, but I can take it from an organic thing from my own premises. And we know very well, I know very well that I'm not using any fertilizers, any chemicals, only organic. So recently I did innovation, I mean a small what you call renovation. I changed everything. Initially it was like this, where there was leakage on my roof. So I want my building to be strong enough. So what I did is I constructed like this. I raised uh, iron railings about 1.5 feet high so that the roof will not be damaged. And I was using coco peat as well as these bags so that it will not be heavy for my building. And also I do composting of my kitchen waste. No kitchen waste, fruit peels will not go into the general garbage at all. It will go only as a fertilizer to my plants. So using organic fertilizers can be a very, very important uh, habit. Instead of throwing it out, you can use it as a fertilizer, especially organic fertilizer, because it is renewable, biodegradable, and environmental friendly and sustainable. So you can make your own homemade fertilizer using fruit skins and peel vegetables, using renewable organic waste. And here I never use any chemicals. My pesticide is neem oil, neem cake, and pungan cake. So all these are small changes which we need to understand. So the environmental thing will not go without the saying of this Swedish scientist. You can see Greta Thunberg, a 17-year-old activist who said that adults keep on saying, oh, it to the young people to give them hope, but I don't want your hope. I don't want you to be hopeful. I want you to panic. That means what? Understand what is happening. It's already fire. So you must take measures instead of creating a hope. I want you to feel the fear I feel every day. I want you to act. I want you to act as you would be in a crisis. Yes, now we are in a crisis. What are the measures we'll take in an emergency situation? We need to take it. So this is a wonderful saying by Greta Thunberg, which we need to understand. So thanks. Any questions, you can find me in the following email IDs. And thanks for your patience. I hope I covered all the things, especially my aim was to tell you only about the health impacts. I hope I covered that. Credits, my special thanks to the management, organizing team, faculty, and the students of Avinashilingam for home science and higher education for women, and all dear participants. And my special thanks to our Dr. Anita Subhash, head of the department, biochemistry, for introducing me, and to Dr. Yamuna for coordinating with me. Thank you all.
Thank you, ma'am, for the excellent presentation.
Thank you, ma'am. Now the session is open for discussion. If any of the participants have any doubt or you want to discuss something, you can either post it on the chat box or you can unmute yourself and please ask the questions.
Good morning, ma'am.
Yes, good morning, ma.
Ma'am, you were told that thermal stress can cause mental illness.
Definitely.
In what way, ma'am, they will cause any disorders, or in what way they cause, ma'am?
No, it is not that disorders. Heat causes some metabolic changes, and as a result, there will be an impact in your brain and your nervous system, which causes some type of stress, and you feel depressed. See, for example, when you go out in a very hot climate, you don't even take your umbrella. What happens as soon as you reach home, you will not able to even speak. You want to drink water and you feel fatigued and tired. For some few minutes that you go out and come in a hot weather, you feel like that. Imagine your body that you are exposing yourself continuously. So there can be even damage in your DNA molecule also. So we do not know. We all know that mutation can happen in our DNA molecules. So thermal stress can be one of the factor in that case.
Okay, thank you, ma'am.
Thank you, ma.
Thank you, ma'am. Does Dr. Rekha Diokar has a question?
Yes, Dr. Rekha, good morning. Dr. Rekha, I can hear you. Dr. Yamuna, am I audible?
Ma'am, you are audible, ma'am. I think by mistake she would have pressed that button, ma'am.
Okay, no problem.
Ma'am, it was a wonderful session, ma'am.
Thank you, Dr. Yamuna.
Thank you, ma'am. Thank you so much.
And all the HODs here concerned. I saw zoology HOD, ma'am. Thank you for all
I
providing.
have one question, ma'am. I am Dr. Manimegle from HOD of Zoology.
Yes,
Ma'am,
madam.
have you published any paper regarding climate change due to vector-borne diseases or mosquitoes?
No, madam. I'm thinking about that because I'm a person working on only foot ulcers, diabetes, and artificial intelligence products and machine learning. and on bioprinting, etc. I was thinking about this. It's a long time thinking, but not yet I have joined. I should work with someone who is good in that, but I can give clear ideas how it happens, because we recently had dengue diseases, and we saw many different changes that is happening in the cytokines, etc. So I think that is a wonderful avenue where we can work on.
Yes, ma'am. My field of specialization is on mosquitoes only. I have
Oh.
completed 5 PhDs, more than 47 papers I have published national and international level regarding mosquitoes only.
Oh,
I
great.
also submitted DST, Department of Science and Technology, UGC,
Okay.
New Delhi.
Okay.
CRT more than 1 lakh 30, 1.5 course submitted the proposal.
Okay,
This month
madam.
last they will publish. We will have the collaborative research, ma'am.
Sure. And it should be, see, whatever research we are working on, madam, my aim is to bring it to the community and should be beneficial to the end user.
Yes, ma'am. Thank you, ma'am. Excellent presentation, ma'am.
Thank you, madam. Thank you, madam. Yes. Madam, please get my number from Dr. Yamuna.
Yeah, sure. I'll talk to you, ma'am. Next week I will talk to you.
Yeah.
We will discuss with our Chancellor and Vice Chancellor. Let me know,
Yes.
ma'am. We will send proposal.
Okay. Okay, ma'am.
Research we will send for funding.
Really, we can do it, ma'am. It's one part we need to touch on vector-borne diseases, especially on mosquito. It's not, malaria is not gone, it is there, and it is not eradicated. We still need to work on that. Dr. Yamuna, is there any queries, ma'am?
Dr. Rekha, do you have any queries?
Ma'am, one more, ma'am. Dr. Subhashini, she is working in computer science department. Now she is working on artificial intelligence, that is emerging technology. We three join together, we will send the proposal, ma'am.
Okay. We are working on AI products. We are working on, because we have a team of AI people also. And in the healthcare sector, we are working on apps and softwares, ma'am. Please do contact me, ma'am.
Yes, ma'am. We will do it, ma'am. We will send it.
Okay, ma'am.
Thank you, ma'am.
Thank you,
Excellent
ma'am.
presentation, ma'am. You was
so nice, ma'am. Thank you, ma'am.
Participants, is there any queries, please? Dr. Rekha, do you have any query? I think there is no more queries, I think, ma'am.
Yes, ma'am. Then thank you, ma'am. Thank you, and I should thank all the organizing team for giving me. Unless I get an opportunity, I have no way of sharing my expertise thing to you. So I have to thank all of you all for giving me an opportunity. So thank you, Dr. Anita and Dr. Yamuna and zoology madam. We'll definitely will work together. When there is a teamwork, I think we can achieve a lot.
yes, ma'am. Yes.
Thank you, ma'am.
Thank you. Can I log out, Dr. Yamuna? Is there anything else?
Ma'am, one moment, please, ma'am.
Yes, ma'am.
Yes. Thank you, ma'am. Your presentation really informed us about the extent of the effects of the air pollution and the resulting climatic change. The facts and statistics you presented were alarming and educating as well. Your sustainable life practices inspires us to follow the same. Thank you so much for sharing your commendable knowledge with us. Thank you so much.
Thank you, ma. Thank you all, dear participants. I have given my email IDs. Once I will just I'm writing it. Please do contact me for any queries, any health related programs, and especially regarding women and children. And as a mother of a special need child, I'm also working with the special need children. And there are so many things I'm working on simultaneously. When we work together, I think we can do service to the human mankind. My email ID is drsheelasasi@sslcareedu.com. Please do contact me. I will readily address your queries. Thank you.
Thank you, ma'am.
Thank you all. Thank you all, ma. Thank you all, ma.
participants are requested to wait. The next session is about to start shortly.
Sabha? Yes, ma'am. Please, ma'am. Yamuna, ma'am. Jayabharti,
Good
ma'am,
morning, ma'am.
already joined, ma'am, in her personal ID, NB Mahalakshmi, ma'am. She joined already. The next.
Ma'am, welcome, Jai Bharati, ma'am. Shall we start the session, ma'am?
Yes, ma'am. Yes, ma'am.
1 minute, ma'am.
Our next speaker for the day, Dr. K. Jayabharti, is the Vice Principal of PPG Institute of Health Sciences, Coimbatore. She has completed BSc Nursing from RBS College of Nursing, Coimbatore in the year 2000. She completed her MSc Nursing from PPG Institute of Health Sciences, Coimbatore in the year 2006 and completed PhD in Nursing from Techno Global University, Shillong, Meghalaya in the year 2003. She started her career as lecturer in PPG Institute of Health Sciences, Coimbatore, from 2001 to till date, and she was promoted as professor and head in the Department of Child Health Nursing in the year 2014. She has been elevated as vice principal from the year 2021. She is now Dr. MGR Medical University Recognized Guide from 2020. Two students have completed their PhD under her guidance. She was a gold medalist in MSc Nursing. She conducted more than 10 conferences and events through SME. She has published more than 15 papers to various international and national nursing journals. She holds position as an inspector in the Tamil Nadu Dr. MGR Medical University, Chennai, Tamil Nadu Nursing Council, Indian Nursing Council, New Delhi. It's a pleasure to have you here, ma'am. The stage is all yours.
Yes, ma'am. Thank you. Am I audible, ma'am?
Yes, ma'am.
Okay. Sabha, can you share this? Yes, ma'am. Doing, ma'am. Doing, ma'am. Okay. So, shall I close my video? Yes, ma'am. Yes. Sabha screen. One minute, ma'am. 1 minute. Yes.
Ma'am, is it visible, ma'am?
Yes,
Yes,
ma'am.
yes.
It's not
Yes,
slide.
Yes, ma'am. Yes, ma'am.
Yeah.
Can you put it in slide share? Slide show?
Yes, ma'am.
Yeah. We can see some.
Yes.
Yes.
So, warm good afternoon, one and all present in this online webinar. Thank you for all the me to take the class of this program, ma'am. Once again, thank for all the people. So now I start up this research design. So next slide. So research design is a vast area. So little bit it may get for it is a you may get for some disturb in your thinking, but it is not like that. Research design is like your story, what you are, how you are taking care of your womb, inside of the womb in your baby, the uterus. So how you are taking care of your child, the fetus. So like that, it's a research design. How you are going to design, how you are going to do for your study. So in which are the way you are going to do for the data collection method, and what type of hypothesis you are going to formulate, and everything you can design and confirmation get by the research design one. Okay. So the introduction. Okay, definition. It's okay, no problem. Definition. See, a blueprint for the conduct of a study that maximizes control over factors that would interfere with the study's desired outcomes is according to the non-CB. And according to the Gothari, the arrangement of conditions for the collection and analysis of data in a manner that aims to combine relevance to the research purpose with economy in procedure. Next slide. So next we can see for what is the purpose of the research design. So it's attempting to find an answer, and it may a big bone, it's a backbone of the research, our research, and it is organized one. So what research methodology we are going to plan, everything, it is a hypothesis and validity and dependent variable, everything we can organizely one, it is bringing into the research design by the research design. And it may give for the implementation and data collection, and it helping for the safeguard by the researchers and minimize the time and money and eliminates the bias and marginal error what will happen in a experimental study, and selection of appropriate tools and advanced planning can happen in here, and possible explanations. So what we can expect, our very confident enough of the explanations we can give through of the this research designs. Next slide. And some features of the good research designs. So that it's a flexible one. The research design is a flexible one according to the experimental and non-experimental, and it is appropriate and efficient and economical one. It should give the smallest experimental error and high reliability and validity by the research design. And others is the 5 important elements, that is the subjects, variables, time, setting, and investigators role. This is a very, very important of the 5 element role. So this is supposed to be in our research designs. When we are planning of our research design, we have to consider these 5 elements. Yes. Next slide. So what made the classification of the research designs? It is a non-experimental and experimental. The qualitative and quantitative is there. In that it is a non-experimental and experimental. In non-experimental, that is a descriptive, comparative, ex post facto, and correlational research design is there. The same thing it is in a experimental research design, that is in a true experimental and quasi-experimental and pre-experimental design. So in true experimental design, that is pre-test, post-test with control, and post-test only with control group and Solomon core group, and factorial design and crossover design and randomized block designs in coming under the true experimental design. In quasi-experimental, that is a non-equivalent control group and time series and multiple time series is a quasi-experimental research design. It is in a one group post-test static group comparison and one group pre-test post-test in a pre-experimental research designs. So we will see one by one it is in a what it is coming, how we have to see for the characteristics, how we are supposed to formulate of the research design we can see. Okay. In experimental research design, method is an empirical research method. It used to examine a hypothesis causal relationship between independent and dependent variables. The next slide. So here there is a three main areas in two experimental, that is a manipulation, control and randomization. So those are done for already. It is in experimental research designs. You may know of how we can able to manipulate of our samples and our studies. We know that. So it is a manipulation, that is experimental does something to at least some subjects. And control, it is experimental introduces the controls over the experimental situation, including use of a control group. In randomization, the experimenter assigns subjects to a control or experimental group on a random biases. So in manipulation, so under the manipulation, it involves the doing something to study participants, that is independent variables. So it may helping in further doing of the manipulation independent variables, that is an experimental treatment, so that what we are giving the management interventions. Okay, and vary the independent variables and observe the effect on the dependent variables, that is a cause and effect we can see. So for example of that one, it is explained, it is a gentle massage is effective as a pain relief measures for elderly nursing home residents. So if it's independent variables, is a gently massage, it could be manipulated by giving some patients the massage interventions and withholding it for the others. Then it is possible to compare the pain level, that is a dependent variable. So the pain level is a dependent variable in the two group to see if difference in the recipient of the intervention and resulted in degree of pain levels. So what are the things we can see it in when you are formulating of the experimental research designs? What is the protocol? That is, what is the interventions we are going to give, and how does it differ from the usual methods of the care? And if there are two alternative interventions, which one is the exactly we can do it, which one is the best for the particular babies or the particular sample? What are the specific procedures to be used with those receiving the intervention, and what is the dosage or intensity of the intervention? So the intervention is that is identical intervention and patient-centered intervention and tailored intervention is there. So the next one is a control. So how we can control here, it is in using in our research design in experimental group, that is achieved by manipulating, randomizing of our sample and carefully preparing the experimental protocols. According to our convenience, we can prepare in some, according to the environment, the experimental environment, we can change for our protocols, carefully we can modify it and we can prepare it. And using in a control group also we can go for this control. So for example, if a researcher wanted to supplement diet of the low birth weight babies with a particular nutrient for 2 weeks, so the weight gain at the end of the 2 weeks would tell us nothing about the treatment effectiveness, because so each month, each day it may gain of the baby weight. So but if you are giving of the supplement also, again if the weight is not gained, so there is no improvement in that. So it won't be given of the any effectiveness of the particular treatment. So it's a suppose on average 1 kg weight gain is noted in that. So does this gain support the conclusion that in independent variables and dependent variables? And control group, so the group of subjects whose performance on a dependent variable is used to evaluate the performance of the experimental group. That is, in our experimental group is in which group we are going to give off the intervention. It's a treatment, that is called as a treatment group. So experimental group on the same dependent variables. So in control conditions, here it is in a control group. So how we can do of it's the control conditions we can put for our research designs. The control group condition used as a basis of comparison in a study is referred to as the counterfactual. And alternative interventions we can do in a control conditions. We can keep it off in our studies. And placebo or pseudo intervention, that means it's there is no any intervention. That is called the placebo or pseudo intervention. Simply what it is, regular routine work is carry over in a particular problem. So that is called the placebo or pseudo intervention. And standard methods of the care and different doses or intensities of the treatment and delayed any treatment, that also is coming under the control conditions. In randomization, there is in some of the phases also is that is in a placing subject in a groups at a random, and random is every subject has an equal chance of being assigned to any group, that is experimental group or control group. So in random, and there is no systematic bias in the group with respect to attributes that would affect the dependent variables. So some examples of this one, it is the effectiveness of contraceptive health education program for multi-para women. So there is two groups of subjects were included. It is one group was given in the intervention, others was we are not giving for any intervention. So in the one group we are giving health education, that is an intervention in coming under the experimental group. So another control group we are not giving for any interventions. So that type of two groups is there. So the women in the sample are likely to differ in the age, education and aptitude. So this would affect the woman's diligence in practicing the contraception method. And some of the methods of randomization we supposed to do for the randomization, how we have to do for the randomization, how to select up the randomizations for our samples. So the flipping a coin and matching, maybe it is in a age wise, we can match for the control group and experimental group, or it is in same educations like that, we can have same economical factors, we can match it. And draw a lots, it's a pulling a name from a cat, and table of a random numbers and cluster randomizations, and reduces possibility of contaminations, and computer generated random numbers, and randomizations.com we can select it for the using of the method of randomization of our sample. So some procedures how we are supposed to select it of the randomization procedure. It is allocation of the concealment and snows and randomization variance, that's a cluster randomization, and grouply togetherly we can take of that, and permitted black randomization and randomized consent or Zelen design, and partially randomized patient frequency, that is a PRPP. So how we are doing of the PRPP is it's a blinding or masking, it is a closing of the one sample, maybe it is absence of blinding, and it may giving of the different bias when we are collecting of the using of the experimental designs. When we are doing of here, it is a performance bias may occur in these conditions sometimes. And randomization.com, here there is a three bases is there, I told already. It is the first one, that is a first generator randomizes each subject to a single treatment by using the method of randomly permitted blacks, and the second generator creates the random permission of treatment for situations where subjects are to receive all of the treatments in random order, and the third generator it generates a random permission of integers. This is particularly useful for selecting a sample without replacement. And some of the independent variables control over how it may taking place in a experimental research design. So that is of the experimental, in quasi-experimental, pre-experimental and non-experimental, how it is taking of the control over. You will see it one by one. So if there is any intervention, we are selecting of the two groups. If there is any intervention, if there is no intervention, that is called it's a non-experimental research design. That maybe it's a descriptive research, or it is any correlational like that it may come, or it is a survey like that it may come. If we are going to give for any intervention particular, the group, so it's the random assignment to treatment groups, that is yes means that is called the experimental research. So again, is there is an intervention? If it is yes, so is there random assignment to treatment the groups? If it is no, or the efforts to compensate for the lack of random assignment. Okay, so there is again between under the experimental research designs, again they are doing of the lack of random assignment. If it is no means that is coming under the pre-experimental research. If it is yes means it's coming under the quasi-experimental research. Okay, by the time, sorry, the previous slide. By the time is the random assignment to treatment the groups, that means experimental group in the group we are giving of any intervention, that is called the experimental research design. Yes, next slide. Okay. So what was the basic experimental research designs will come? That is a pre-test, post-test designs control group. Usually we mostly we will follow for this pre-test, post-test design control group. And next is the sometimes we are using it's a post-test 1D experimental design and Solomon 4 group designs and factorial design and randomized block design and crossover design, that is repeated measure designs we are following. So if it is a what it is X, before going off all the symbolic representations, what we suppose know that is an X is denote the experimental manipulation, that is giving of the treatment or it is interventions, and 0 for it is a observations or for it is a randomizations. Because always our methodology we are using of that randomization, that symbols only maximum we are using. Okay, in pre-test is next slide. In pre-test, post-test control group design, that is in before and after design, that is classic experimental design. It may use in a commonly all the times, and 2 randomized groups we will have, and 1 group receive the experimental treatment, another one we are not giving any treatment, that is called the placebo treatment. So that is a control group. This is the pre-test, post-test design. Here there is a denomination, it is a denote of it's given, it's a randomization. So the control group C is a control group, E for experimental group. The pre-test control group is getting of the first observation, and post-test we are doing of that, but we are not giving for any intervention. But in the experimental group we are giving of the intervention. X is we are giving the intervention. For example, it is the effectiveness of antenatal nutrition education on pregnancy and labor outcome among primary para mothers attending outpatient department of the selected hospitals. This next slide. So this is another method. This is a repeated measures designs. So here we are taking up in experimental designs. Here we are giving up the intervention. So one group, it is a control group. There we are not giving any intervention. The pre-test we are doing, there is no intervention, and after that post-test we are observing of 2 or 3 times we are observing, that is O2, observation 2, observation 3 and observation 4, that is a post-test we are doing of that. But it is the same time in experimental group, pre-test we are doing, after we are giving intervention, X is the intervention, and after that post-test we are doing observation 2 and 3 and 4, we are repeatedly we are measuring of the using of this design. Maybe if the eye fluids getting of any baby, we can assess the fear and anxiety for the baby. In this time also, before that we have to do for how much the fear and anxiety is that baby, and after giving the instruments, interventions like for some diversional therapy for the baby, and after diversional therapies, we are checking of the immediate 10 minutes how much of the fear and anxiety of the baby, after 15 to 20 minutes how much it is was, and after half an hour or after 1 hour how much the fear and anxiety the babies have. Like that we can assess the baby, for we can use of this repeated measures designs. And post-test only control group design, it's after only design. Here it is a one group we are giving of the only we are giving of the intervention for one group and doing of the post-test. So the another group, in control group, we are not at all doing of the any even pre-test also we are not doing. Only we are conducting of the post-test and we are getting of the score and we are analyzing of the. And for example, it is the effects of holding the newborns and paternal bonding behavior. The researcher randomly assigned 36 first-time fathers attending the uncomplicated deliveries, that is a normal delivery of the normal infant, to experimental group and control group. In 12 to 36 hours after the babies were born, bonding behavior frequencies were recorded and observed the outcome in this study. In Solomon 4 group designs, that is involves 2 experimental group and 2 control groups is there. Here it's a 1 experimental, 1 control group or administered pre-test, and other groups are not having of that, and allows the effects of pre-test measure and interventions to be segregated. If you are seeing that symbolic representation, you can get for the ideas. In Solomon 4 group designs, there is a 2 control group, 2 experimental group. Here there is one control and one experimental is not having of the pre-test. But the experimental group is having of the intervention and they are having of the posters. Okay, so in Solomon 4 group design, here we are having of the 4 group of the people, 4 group of the sampling, but one control group, one experimental group, not at all having of the pre-test. Only one control, one experimental having of the pre-test. Yes. An example for an effectiveness of workshop on management of HIV to improve the nurses' attitude toward patients with AIDS admitted in a selected hospitals. Here in this study, here we can use of the Salomon 4-type SAP. So this is another area. It is a same thing is like in a Salomon 4. It is used to manipulate 2 or more independent variables simultaneously. Here it is independent variable, the factors in each factor levels 2 or more, and treatment conditions we can be for it's like in a group. So design it may be in a 2 into 2 and 2 to 3 or 3 to 3 or 3 to 4 groups we can have for it's dividing a sample group and we are conducting of the giving the interventions. So for example, it is a one group we are one side we are doing of it's a type of stimulations and another one we are seeing for the daily exposures, how it may be affect of that. So we are giving of it is an auditory and tactile stimulations we are giving of the group. Here it is a A1 and B1 we are mixing and we are checking with the through interventions. After the interventions we are checking of the auditory and the interventions, and after that again we are checking of the tactile plus interventions, how it may be elicited for the what is the outcome is what we happen we are checking. So like that, and B2 and after the 30 minutes and after 45 minutes how it may exposure of the particular patients have due to of these stimulations, how they are getting of the outcomes we are checking. And this next one, randomized black designs. Here this is similar to factorial design, and it may be in a 2 factors maybe will here and 1 factor not experimentally manipulated, it may blacking in a variables. So sometimes unknowingly we are doing of the treatment, but the patient is not knowing, the sample is not knowing of the management. And designs is a 2 into 2 and 2 to 3 and 2 into 2 into 2. It is a comparison. The example is a compare the effect of tactile versus auditory stimulation for male and female infants child in a ward setup. So it is like a example. So here they are comparing of the gender. It's a male and female. Like it's the same thing. It's a type of stimulations with the auditory. So the both genders they are involving here. They are specifically they want to take off. It is a this research designs male this much in we are taking it in the experimental. The same thing we have to take it in a control group also. Yes. And crossover design, this may be a repeated measure or counterbalance designs. So sometimes it may be in a carry over, it is things in within the group itself it may getting of the some carry over of the effect of the particular sample group design. It involves the exposure of the same subject to more than one experimental treatment again and again it may appear, and the subjects that is or the sample are randomly assigned to different orderings of the treatment, and extremely powerful design but inappropriate for certain types of research due to the problem of carryover effects. So like this is maybe it is effectiveness of 2 alternative backrest position, maybe it is in a flat or horizontal versus 30-degree elevation. So here we are mentioning only one thing, it's a flat versus 30-degree elevation. But here it's a crossover effect may occur. So here that is a flat and horizontal. It may included here and versus of the 30-degree elevation. So it's on intracranial and cerebral perfusion pressures in a brain injured adults. We are using of we can use of this type of research design and we can elicit the outcome. So what was the experimental design strength? So how we can get it of the experimental research, when we are doing of the experimental research designs, we suppose know for some of the strengths, some weakness also is there. So firstly, you see the stent. It's a powerful method for testing hypothesis of cause and effect relationship, and it's the highest quality evidence regarding effects of specific interventions. Maybe if then the relationship important because of implications for prediction and control in our experimental studies, and strengthen the lies in confidence with which causal relationship is inferred. So another the experimental strength, that is through the control, it's imposed by the manipulation, comparison and randomization, and alternative explanation can often be ruled out in our sample. When you are selecting the sample, when you are doing of the data collection, or what else happen in the during the research environment, we can do of the according to our convenience and the environment of the research, we can change it. So that also is giving of the strength of the particular research studies, experimental research. And sometimes it may be limitations. So always we should have for the strength and weakness in limitations here, impractical or impossible may happen this research designs, this experimental and RCT. So the randomization control, it may happen, and in this clinical setting, that is randomization control trial, clinical settings have get for the using in a clinical staff alone than the researchers. So it needs biggest area in the RCT areas, how they are doing of the trial and error method in medications. So it can be difficult to determine the intervention group actually received treatment, and if the treatment group did not. So the other limitation is here, it is difficult to maintain the integrity of the intervention and control conditions if study extends over time and experimenter does not have control over the clinical environment, and treatment may be diluted through the not participating in a group. Maybe in one ward we are taking for any infant, so alternative one another room 1, 3 and 5 we are taking for the babies, we are checking the height and weight, and we are doing for some intervention by that group, and 2, 4, 6 we are not doing of that same infant age of the baby, but we are not doing of any treatment. Maybe they may discuss in that area and they may dilute here. Okay. And heart thrown effect may occur. So that means the heart thrown effect means inclination of people who are the subject of an experimental study. So that means, for example, if a child is doing for any mistake, the mother has to be tell for the child not to do of that. So always we can do, always we can tell to the child. But if some others are telling the mistake on the child, if the own mother is not accepting, and mothers will do of the inclination behavior. So that is she will accept and she will tell for, she won't change her behavior, she will accept it of that. So here, so the inclination of people who are the subject of an experimental study to change or improve the behavior being evaluated only because it is being studied and not because of changes is the experiment parameters of the stimulus. Okay, the next slide. So next one is the quasi-experimental. So the quasi-experimental research design, it is a research design in which the researcher initiates on experimental treatment, but some characteristics of true experiment is lacking. In quasi-experiments, that is, it's involved the intervention but lack of the randomization. Already we done it for our research, we all know that, and facilitate search for knowledges and examination of the causality in situation in which complete control is not possible. Here, in quasi-experiment, what it is that only we can explore. In types of quasi-experimental design, that is a non-equivalent control group design or interrupted time series design we can see. So in one group posters, only design we can use. Here the treatment, we are giving the treatment, the baby came for the pneumonia, so we are giving the treatment. After that we are doing of the evaluation, that is a posters, and we have to check what is the outcome of the treatment of the pneumonia baby. Okay. In non-equivalent control group posters, only design that is pre-experimental. Here we are giving of the intervention, we are giving the treatment, we are giving experiments, we are showing for some of the interventions, and we are doing of the posters. The another one we are not doing of the intervention, simply we are doing of the posters. So for example, it is introducing a computer education to improve the staffs. And some of other example that maybe it's a quality assurance model we can give for one hospital and the staffs to impact on the job satisfaction of the nurses, and use here the job satisfaction nurses in another similar hospital as a comparison we can check it. If two groups are similar on pre-test, we could be relatively confident that the difference in posters is the result of the intervention, because we are teaching of the something in the one group. And quasi-experimental in the term comparison group is used in a lieu of the control group. Lieu means it's instead of that. And one group pre-test posters designs, here it's a pre-test and we are giving up the intervention, and posters we are doing. So for example, it's the effectiveness of after delivery, and we are teaching of the doing of the studies effectiveness of pelvic floor muscle exercise in control of the incontinence of the urine. And one group pre-test posters design. So here it is in a most commonly used the designs for all the UG and students. It seems a straight forward what it is that we can explore, and it has been some weakness in a pre-experimental. It also called the pre-experimental research design. It cannot be controlled the external factors. So for example, here I am giving off it is a anemic child for taller babies. I want to give off it is a Nelakala I want to give for that baby to measure of 1 month continuously. But by the time simultaneously in the home, they are taking off some iron rich diet, they can take it, or they can have for any iron syrup. So in the time, the external factors I cannot control over that. But at the last, my data collection period, if I am checking of the blood test, the same blood volume, the hemoglobin volume is increased. So I cannot say for through my molecular meta only it is giving of the evaluations. It's giving the improvement of the hemoglobin. And next is the time series design, that is interrupted time series. Here the data collected over an extended period, and intervention is introduced, that is X is already we saw that denoted is a intervention, and O1 to O4 that is an observation 2 and observation 4, that's 4 separate instances of data collection, and O5 to O8 that is 4 treatment, post-treatment observation may be there. So for example, it is O1, it is a number of death in a Jan to March in this year before RRT test we can take. And after RRT implementations, the data on mortality are similarly collected for the 4 consecutive of the 3 months. So that is a 01 to 04 and 05 to, sorry, O2, O5 to 08. And time series designs, that is in a, this is observation 1, 2, 3, 4, and we are giving the interventions, and after that, again we are checking of the posters, that is observation 5, 6, 7, 8, that is effectiveness of selected nutritional interventions on growth and development pattern of children attending the 5 clinic in the areas. So this is another one, the time series non-equivalent control group. So here in non-equivalent control group, here is one group we are giving of the interventions, the another one we are not giving the interventions. Okay, so the first group we are giving of the, we didn't give for the equal importance, equal durations, we didn't give for that observation 1, and after little bit take long time, and after we are coming of the observation 2, 3 and 4 we are giving of the equal intervals. So it's non-equivalent. And time series with multiple institutions of the treatment. Here it is a frequently we are giving of the interventions for the particular group. First we are conducting of the observation 1 and observation 2, we are checking it and we are giving the treatment. That means interventions we are giving. After the interventions, after the class, we are checking it is the observation 3 and 4. Again, we are inculcating our interventions to the particular sample group, and again we are doing of its observation 5 and observation 6, and again after that we are doing of the multiple times we are giving of the interventions. And this is for time series with intensified treatment. Sometimes we are giving, sometimes we are not giving, sometimes simply we are observing and we are taking care of the particular sample. So first time we are adding of its observation 1 and observation 2, we are giving the intervention and follow-up of, and we are doing of the outcome, checking the outcome, observation 3 and 4, and again we are giving the interventions 2 times we are giving. Normally 1 time we are giving, additionally 1 time we are adding, and again we are checking of the outcome. And after that, and after the 6th time, again we are giving the normal interventions with the additionally adding of the 2 more times of the intervention. So that is the intensified of the treatment. And time series with withdrawn and reinstituted treatment. So that means this is a normal one. So when we are giving of the 2 observation, we are checking, we are giving treatment, and again we are checking, again we have to do for the 2 more times of the observation. And after the 2 more times, we are not giving of the any interventions, simply again we are doing of the treatment. So we are checking of the assessment, and after that 2 more times only we are doing of the intervention, and after that only we are checking the last one. So in between we have to do for the intervention, but we are not giving the treatment for the particular group of the samples. And this is a single subject experiments. Here the baseline data gathering, what it is available, maybe if we are getting for any cerebral palsy regarding we are doing for any research on only, it's a rare case, it's a GBS babies or any cerebral palsy babies, it's a what is the baseline data gathering we are getting and we can use it. And intervention basis what it is getting, and that is we are using of the AB and besides we are applying here. So it is sometimes the treatment is withdrawn in here, and sometimes withdrawn the treatment is and reinstituted, that is called the AB designs. Another quasi-experimental research design, that is regression discontinuity design and dose response designs. So here the experimental and comparison conditions here is to develop a strong interventions protocols we are preparing, and what are things we needed, how we can do for the manipulation, where we can do for the control, and everything we are we are formulating protocols and we are doing, and careful documenting the counterfactual of the what it is coming of the outcome we are entering. And in time series design, the counterfactual is the condition existing before the interventions. And this is strength of the quasi-experimental, that is maybe it is a practical aspect may helpful and difficult to conduct the true experiments in nursing research in a real life settings, we can use it, and introduces some research control when full experimental rigor is not possible, and several rival hypotheses competing with the interventions as explanations for the results, and take the weakness into account in interpreting the results. So some of the limitations, already we saw that is a strength, now we will see for the limitations of the quasi-experimental. So there is a several rival hypotheses competing with the intervention as explanation for the result, and take the weaknesses into account in interpreting the result. And conclusion depend in part of human judgment rather than more objective criteria, and cause and effect inferences may be less convincing in the quasi-experimental. So here it is a RCT test. Here they are following of the 1, 2, 3 and 4. It is a basis we are following. So we will see it is a one by one the basis here. So the base one, it is a initial testing for of your new drug that focuses on determining the best drug dose and identifying the safety effects. And base 2, it is a seek preliminary evidence of efficacy and side effects of the drug dose determined by the base 1 trial. And do not use control group or randomization cannot be classified as experiments here. and more similar to the pilot studies we can use. And the base 3, that is comparative definitive studies in which new drugs effects are compared to those of the drugs considered, that is in a standard therapy, what we can follow it in the clinical setup. So the full scale definitive clinical trials we are following here, and sample size not determined before the data collections. How much it's a sample is coming, we can use it and we can add it up to our data collection and data analysis at intervals to test for significance between the groups, and data collections is discontinued if significant difference is found. So the base 4, here is the trials occur after regulatory approval of the drug and designed to follow patients over time to identify uncommon side effects and test marketing strategies will happen, and do not include a control group or randomizations. And some of the expectations of the CT, that is in nursing field, it is a base 3 trial. It is designed to be definitive test of the hypothesis we are supposed to formulate and evidence available from previous studies that intervention causes the desired outcomes. We have to follow that and we can make it in the evidence base, and clearly define interventions and protocol established for clinical applications and study conducted in a clinical setting only, and experimental designs used here and inclusion exclusion criteria. It should be not definitely what it is, like that only we can use it. We can't change and we can manipulate it after committing of the studies, or during the studies also we can manipulate, and baseline states comparable and all study groups included. And expectations of the clinical trials I think it's selected subjects randomly assigned to treatment and control groups, and success secured individually over time as they center in the clinical areas, and study has high internal validity, that's a rigorous high level of control and bias rule out, and blinding and double blinding we can use. So the blinding may be what the treatment is going for the patient, maybe the patient is not knowing of that. In the double blinding, sometimes the patient and researcher is not knowing of, simply they are giving the treatment, the interventions, it's a double blinding may happen. And treatment is equal and consistently applied to all subjects in a experimental group. And expectations of clinical trials in the nursing, the remaining thing is different variables are measured consistently and designed approved by external experts, and adequate funding to allow rigorous design and adequate sample size, and application of findings in a clinical practice and interventions defined in detail so clinical applications can be achieved. So next we are moving on to the non-experimental research designs, that is a qualitative. So here the qualitative research design, it is originated in the discipline. Already we discussed for the experimental research, then this is a qualitative, and non-experimental, it's anthropology and sociology, but is now used to study many psychological topics as well as they are following of this one qualitative study. It is generally begin with the less focused research questions and collect the large amounts of relatively unfiltered data they are collected in a public, and they can produce of the result. And the definition of the qualitative research is a research method used extensively by scientists and researchers studying human behavior, opinions, themes, and motivations. And some goals of the qualitative research, it seeks a depth rather than the breadth, and instead of the drawing from large representative sample of population, qualitative research seek to acquire in-depth and intimate information about the smaller group of persons. And to learn about how and why people behave like this, it's a particular big area. So we can do for this qualitative study, think and make a meaning as they do rather than focusing on what people do or believe in a large scale. So the goal falls within the context of discovery rather than verifications. So some types of the non-experimental research design, that is a historical research and phenomenological and ethnography, grounded theory and secondary analysis and meta-analysis and case study and action research and content analysis and field research is there in a non-experimental, and some of the things we can have for it. So in historical, it's in name itself, we all can able to identify it's a historical research design. It method refers to the use of primary historical data to answer a question. And because the nature of data depends on the question being asked, and data may include the demographic records such as birth and death certificates and newspapers, articles, letters and diaries, government records, or even its architectural drawings. And some characteristics we'll see that it involves careful study and analysis of data about the past events, what was happened in previously we can discuss. It is critical investigation of events, the development and experience of the past, careful weighing of the evidence and interpretation of the weighed evidence. So for example, nowadays we are in a corona period. So in what was happened in the 2020 March, April. So at present also now we can think and we can correlate of the things. Yes, next slide. And next method is a phenomenological method that is explore how people is taken for granted, world is experienced, and how structures of consciousness appear in the world. So the definition of it is an approach of philosophy that begins with an exploration of a phenomenon, logical, ontological, metaphysical spirit that is behind the phenomenon. This is named as a dialectical phenomenology. So according to G.W.H. Hegel. So this is the outcome of this method. It's a full rich description of a human experience, what has happened in the particular time. So that we can get it of the experience we can get it of the answer, the outcome of this method. That is a live experience of the study, and take participant descriptions of the live experience and read all the participant description about the live experience in the particular situations. Next slide. And characteristics of phenomenology tends to withstand the acceptance of those circumstances which are unobservable and is grand system acted in a speculative thinking. And phenomenology opposes naturalism, and that is subjectivism and positivism. What has happened, that only we can say. Yes. So the data collection, how they will do in a phenomenology, that is an in-depth of interviews and diaries and other written materials. We are through that we will collect it and they will submit. It's a task of contrasting and comparing the final data to determine what patterns and what are the themes is occur. The investigator seeks a further knowledge about the live experience in a conscious manner. Is the next slide. So the sum of the types of phenomenology, that is realistic, existential, and constitutive and hermeneutical. So in realistic, that is the research focuses on a gathering the universal abstract of various types of information, including the human action, motives, and result. The next slide. So it's another one type. It is a constitutive. This includes philosophy of the natural science. So here the procedure entangles suspending acceptances of the pre-given position of the conscious life as something that exists in the world, and carried out in order to obtain a ultimate intersubjective grounding for the world and positive science of the thing. So that is a social belief what we are following in our particular area. Yes, next slide. So existential method, that is a research design. Here it is a study is concerned with the topics such as the action and conflict, desires, finitude, and oppressions and death. And next, hermeneutical phenomenology. It is the uses of the lived experience as a tool for better understanding of the social, cultural, political, or historical event. So, for example, its phenomenology study on lived experience of tsunami victims in a selected village of Tamil Nadu. So, next one is ethnography. It is a nature of the specific social phenomenon, and often using a small number of cases, we can make it a sample and we can get it out. And ethnography emphasizes the observation of details every day of the life and naturally unfold the real world. Next one. So the characteristics of it is ethnographers learn about the cultural groups, what is happening in the particular society, so that they are interested to learn through the extensive field work. So it is labor intensive and time consuming endeavor where even month of the years or field work can be involved. And you can go for the next slide. So the ethnographers rely on various sources of the data collection through of the in-depth interviews, record analysis, and observation of the physical evidence, photographs, diaries, letters, etc. Next is a step that is a free field work and field work base 1 and field work base 2 and field work base 3 and post work field work, like that they will do of the data collection thing. Next. So this is this. Next, there is two types, that is a macro and micro. So in macro, it is a study of broadly defining the culture, and micro they are more have for the more narrow aspects of the particular what is happening inside of the culture. So the example it is, you can go for it is a cultural belief of peoples about the antenatal, natal, and postnatal care for women in selected tribal communities. So next is a grounded theory. So the theory uses the interplay between the analysis and data collection of the produce the theory. You can go for example. So it is an so the overview of the basis here, it's a data collection they will do for it some note taking, and after the each hour of the data collection, see each and everything look and corner they will correct it of the points and they will do for finding of the outcomes. And next one. Next slide. No, next slide. The secondary analysis. Here it is we know that it is not the first one, it is the next secondary analysis. We can go on the search of the content in a second way, that is in a research method. It saves both time and money and avoid unnecessary duplication of the research effort. The second analysis is usually contrasted with the primary analysis, which is analysis of the primary data. It's collected by a researcher. So they can go for it is in the examples of secondary thing. It's a textbook, encyclopedias, and new articles, review articles, and meta-analysis. They can do it. Yes. The best technique it is collecting of the information. Yes. Next one. It's a meta-analysis. It is a technique that uses the findings from several studies. So it creates a data set that may be analyzed as a single piece of the data. So by applying the statistical procedures to this finding, these techniques can be a unique approach to integrate the knowledges. Next is a field research, that is a natural environment, what it is happened in the particular field, the tsunami area, what has happened at the time. So we can get it of the information. It's implemented control or experimental conditions to pick up the such methodologies are especially useful in observing the social phenomenon over the times. So field research, you know that it is in particular the field they have to go on approach in each and every part they have to assess. And case study method, it is a descriptive or exploratory or explanatory. They can do it in a case study method. And survey method also they can get it of that. And case it's a studies of specific bounded system, that is a person or institutions. And this one is able to replicate and Hawthorne effect and researcher bias and time intensive and possibility of errors and ethical issues may happen in a disadvantage of this method. Next slide. So action research, that is action research, it's inquiry or research of context and focus efforts to improve the quality of the practice. So maybe if they are having some problem, we are finding of the particular areas, and when we are collecting of the information through data collection, again we are giving the interventions and making into the clear of the particular thing. So that is called the action research. It's typically designed and conducted by them. So the method is interview, observation, and storytelling and social drama, like that we are doing of the data collection method of the action research. So this is cyclic process of the action research. an objectives, yeah. Action research is of a particular importance to the nursing field, especially in the Indian setup, as its objectives are followed. It's to solve problem by enriching the field of application of a discipline. So the last one, it is a content analysis of the research design. The content analysis is used to analyze the presence, what it is existing, and meaning and relationship of words and concept in a text, then make inference about the messages within the text. And methods identify the specific body of material to be studied, and small amount of material is usually studied in its entirety. Yes. And role of researcher in non-experimental research design, here it physically go to the people, location, settings or site. They have to find it of the outcomes. Here the researcher cannot do for any manipulation in the situations. Yes. So the researchers, the strength and weakness, it is a in-depth of understanding and flexibility of it is a non-experimental research design order. Strength and weakness is a subjectivity and suggestive, not definitely we can follow, and limited generalizability only it is there. So that's all about of the research designs of the experimental and non-experimental research designs. So I think you may get for some of the information in this research designs. Thank you for the patient listening of at this lunch time. Thank you for your patient listening.
Thank you, ma'am. Now the session is open for any queries and discussion.
Okay. Anybody have, ma'am?
Participants, if you have any queries and discussion, you can either unmute yourself or put it in the chat box.
Sabha?
Yes, ma'am.
Okay.
Participants can unmute and ask if you have any questions.
Actually, this research design is very vast area. I know I need to have for more than it is one day to discuss for this research designs. So it's even it's 1 hour. We cannot dedicate for this one for the research designs. Anyway, they gave me for that. So in that I got it for so little bit slides only. I explained of that. So anyway, if you have any doubt, you can ask. Because the research
Ma'am, the session was
Maybe
very
no questions,
informative.
ma'am.
Yes, Sabha.
Ma'am, Yamuna, ma'am, I think no questions.
I think that there is no question seems, ma'am.
Yes, ma'am.
Okay. Dhanushri, proceed, Dhanushri.
Yes, ma'am. Thank you, ma'am. Your session has gave us the basic knowledge about the experimental design in research. It also gave us an insight about the clinical trials that are carried out during a particular research. It taught us a lot about how to perform a research in an organized way. Thank you so much, ma'am, for sharing your knowledge with us. The participants are now requested to take a break for lunch and join back at 1:25 p.m. sharp. Thank you.
Sabha, shall I leave?
Yes,
Thank you,
ma'am. Yes,
ma'am.
ma'am. Yes, ma'am. Wind up, ma'am. Thank you, ma'am. Thank you so much, ma'am.
Thank you.
Ma'am, good afternoon, ma'am. Ji, ma'am.
காயத்ரி காயத்ரி
மேம் சொல்லுங்க.
ஸ்பீக்கர் வந்துட்டாங்களா?
ஜாயின் பண்ண சொல்லிருக்கேங்க மேம். மேம் விஜயகுமாரி மேம் ஜாயின் பண்ணிருக்காங்க.
வந்துச்சா?
விஜயகுமாரி மேம் ஜாயின் பண்ணி இல்லைங்க மேம். விஜயகுமாரி மேம்
எங்க இருக்காங்க? விஜய் மேம். ஓ விஜய் மேம்.
மேம் குட் ஆஃப்டர்நூன் மேம். விஜய் மேம்.
பாஸ் விஜய் மேம். விஜய் மேம் எங்க ஜாயின் பண்ணிட்டு போயிட்டாங்க?
மேம் லைன்ல தான் இருக்காங்க. ஆனா மேம் அன்மியூட் பண்ணல.
சரி எங்கன்னா கிச்சன் கிட்ட போயிருப்பாங்க.
கிரண் வெல்கம். மேம் ஹிரண் ஜாயின் பண்ணியாச்சு.
Hiran ma'am, welcome.
கிரண் பாய்.
கிரண்
எப்படிடா இருக்க? How are you? எப்படி இருக்க?
Hiranmai.
Hello. Good afternoon.
Yes, Hiran. We are able to hear you. Yes. Hiran?
Ma'am, shall we start, ma'am?
Yes, ma. Yes, ma. Yamna?
Yes, ma'am.
விஜயகுமாரி மேம் ஜாயின் பண்ணிருக்காங்க. மேம் விஜயகுமாரி மேம். கிரண் வாய்ஸ் கேட்கல.
एक मिनट आना ऑफिस.
Yes.
A warm afternoon to one and all present here. I take immense pleasure in introducing our guest of the first session, a speaker, Dr. R. Y. Hiranmai, Professor at School of Environment and Sustainable Development, Central University of Gujarat, Gandhinagar, Gujarat. Our resource person, Dr. Hiranya Yadav, after obtaining PhD in Life Sciences from Avinashilingam University for Women, Coimbatore, she has been working as a quality controller in pharmaceutical industry and then started career as an assistant professor at Karpagam University, Coimbatore, followed by teaching and research at School of Natural Resource Management and Environmental Sciences, Haramaya University, Ethiopia, and back to India as assistant professor at Avinashilingam University for Women, Coimbatore. During the career, she is actively involved in research through guidance, projects, presentations, and publications in the field of bioremediation through vermi technology and organic farming to improve soil and crop quality. More emphasis is given to the research related to soil quality, solid waste management through bioconversion, and organic farming. Sustainable practices of waste management and organic farming for soil fertility and crop production is the thrust area of research. She also holds the administrative responsibility of presiding officer, ICC, Central University of Gujarat, Gandhinagar, Gujarat, India. Also actively involved in NSS, EBSB, and academic and extracurricular activities of the university. She has organized many national and international conferences, workshops, and webinars. We welcome you, ma'am.
Thank you, Dhanushri. Am I audible to you now?
Yes, ma'am.
Okay, thank you. Thank you so much for a nice introduction to me. And being an alumni of the... Yes, ma'am. Good afternoon, ma'am.
Good afternoon. How are you, Erin? Right?
Fine, ma'am. Fine. How are you?
Okay.
So, with your permission, I'll start the presentation, ma'am.
You can start now.
Yeah, okay. Is the slides visible to you?
Yes, yes.
Okay. Okay, so full screen. Okay. So very warm good afternoon to all the faculty who have joined this, and a special note of thanks to the HOD of Zoology and HOD of Botany, both of are present here, and Dean Madam Dr. Vijay Lakshmi, and my supervisor Dr. Vijay Kumari ma'am has joined this. So I thank all my teachers, those who have been a part of my life and molded me to this stage. So I can say that it's a pride moment that I'm invited to give a lecture as a resource person on an auspicious day of Earth Hour. So Earth Hour, as we all know, so it's representing the complete Earth today and how we can save the environment as well as the Earth. So, so today the Earth Hour is celebrated through the World Wildlife Organization, which is giving an increased awareness and spark a global conversation on protecting nature. And to tackle the climate crisis, since the topic of our discussion today is on climate change and agriculture, it's a significant day for us. And we all need to work together to shape a brighter future. So from the beginning, the concept is that every year, the last Saturday of March is celebrated, and at 8:30 p.m., we switch off the lights for a moment to give the contribution to support the world and our planet. So with these words, I'll go to my presentations. So the presentation will start with the climate change and agriculture. So what's the need for us to have a climate resilient agriculture systems? So if anybody is not audible or there is any difficulties in the technicality in visibility, you can just tell me at any stage. And the discussion also, if anyone want to ask, you can have that at the stage. So I'm grateful to you for inviting me once again, and we will start with the session as a climate change and agriculture on this most important day of Earth Hour. So the climate and Earth, the evolutionary process that have passed through many generations for decades or millions of years, that has given us an Earth to survive on. As since the researches are being conducted, we know it's the only planet where we find the lives. That means the living organisms are there, and the biodiversity that helps to circulate the nature and also to balance the earth. So presently, due to various activities, we know that the most contributor of this climate change is human being. So to manage our needs or to the future, we need to think about a day when we can think, because the future generation too enjoys this day. So the complete earth is considered as an ecosystem, because in most of we from the sciences we learn the ecosystem and ecology. So considering it into different parts of aquatic or terrestrial ecosystem, and then further into the community or population. So we will consider the whole earth as an ecosystem and how it can be balanced. So coming to the introduction of this topic, we can say the climate system, the most complex and interactive system on the earth, which is affected by various factors. It has the atmosphere, the land surface, snow and ice, oceans and other bodies of water, living things. The Earth's climate is most affected by latitude, tilt of the Earth's axis, movement of the Earth's wind belts, the difference in temperature of land and sea, and the topography. The climate, as we consider the weather and the climate, these two factors we always think and talk about, and how these two are connected. So it's a long-term weather condition of a place, which was including the temperature, humidity, atmospheric pressure, wind, precipitation, etc. So the climate of a place, which will depend on latitude, altitude, terrain, and the presence of nearby water bodies and their currents. So even if we take a place, for example, of Coimbatore, the place had a very pleasant climate some 10 to 20 years before. That's lacking right now, because the temperature never exceeded 30 to 35 degrees, but we can say that presently it's exceeding. And there is a erratic rainfall pattern changes. So added to these, there is more of urbanization. So combinedly, all these are influencing the weather condition that's giving ultimately a change in the climatic pattern. So it's a complex system which is formed as a result of interaction of variable factors: the aquatic ecosystem, terrestrial ecosystem, Earth's environment, atmosphere, glaciers, and most importantly, the living organisms. Because the activity of each and every living organism is influencing the non-living part of it. And all these interactions are ultimately releasing the heat or the variation in temperature that's deciding the climate of a particular place. So we know that globally we are surrounded by most of the water part in oceanic region. So these water currents have a high influence on the climate, because even if it's a hot summer, if there is a cyclone approaching, then the weather pattern changes in that region. And these are all due to the water currents, and a majority of part we know that As per the 2021 survey of the Forest Department of India, you can see that there is only 3% of forest that is left behind. So considering only the real forest part of India, so we have to expect the changes and work or act upon this. So how these air circulations are affecting climate. On one part, we know that the solar energy is continuously coming and the earth rotation is influencing the changes. So in two parts, you can see either the warm air, which is giving to the near equator zone, and their cooler region on the other part, the cold deserts or the hot deserts and the forest side. So all these are together responsible for the climate change that we observe. And these changes in the climatic pattern, along with the anthropogenic activity, is affecting the agriculture of any country across the globe. So mostly we know that agriculture is dependent on rain or the monsoons. So the rainfall pattern, when it changes, it's influencing the agriculture as well. So the prevailing winds pick up moisture from an ocean, and on the other side you can see where there is a wind word which can stop the clouds and give a greenery, and on the other side you can see the air is descending, it's warm and little moisture that reduces the pattern or the land use pattern of the region. So the climate change is not the change that have occurred in a very short duration, but it has took over periods of decades to millions of years due to processes such as the plate tectonics, volcanic activity, thermohaline oceanic circulation, the changes in the ocean currents, rainfall, vegetation types, and coverage density. So all these changes that are occurring are adding to the amount of atmospheric greenhouse gases, and this is resulting into the global warming. So these results will lead to the extreme weather patterns, that's giving you the floods, droughts, or the elevation of temperature and storms. And coming to the climate, it will be affected, associated with both natural and anthropogenic factors. So this has altered in almost 150 years ago, and we need to understand the key factors that are responsible for these changes and try to modify them to develop a better environment for the future. So there are many studies that are going on from the Department of Climate Change or the Ministry of Environment, Forestry, and these studies and also the research organizations of all over the globe, they are also trying to balance the temperature and how to reduce these emissions and manage the climate of the earth. So most of the greenhouse gases, we can say, some of them are released naturally, but most of them are added to the atmosphere nowadays due to the human activity. And reduced forest cover is giving the other tough time to manage the released gases. So the human activities that includes the combustion of fossil fuels in the electricity generation, transportation sectors, agriculture, land use change, waste treatment, and various industrial processes. So all these are adding to the carbon dioxide, methane, and nitrous oxide. So agriculture, we can say that it can have a solution to climate change as well as it was a cause to climate change. So the changes, how it's affecting. So once there is a change in the temperature or the gas patterns, there is a disruption in the relationship between the organisms. That can be either parasitic relation, pathogenic interactions, mutualistic relations. So many activities are there which are interrelated. So here the relation is getting disrupted between the soil, plant, microorganisms that are surviving in a given area. And this will lead to the loss of biodiversity in plants, animals, and microorganisms. The pattern changes in the microbial interaction will lead to changes in the biogeochemical cycle that leads to the reduction in soil. Yeah?
Sorry for the interruption, ma'am, but the slides are not changing, ma'am.
Okay. But for me it was changing. I'll close it and start again. I have shared it. Is it visible now? Hello?
Yes, ma'am.
Okay. Is it changing?
Yes, ma'am.
Okay.
Yes, ma'am. Yes.
So I was here in this slide. Is it visible now?
Yes, ma'am.
Okay,
Yes.
thank you. So we have been discussing about the species or the biodiversity pattern, how it's influenced by the climatic changes. So how these changes in the weather patterns can influence the biodiversity. Because bacterial diversity of the microbial diversity is of significance to any life forms in the earth, either it is in the aquatic ecosystem or in the terrestrial ecosystem. Because they are the major players of bioremediation and biogeochemical cycle. And due to increased tradition of the waste by the human beings, it's unable to recycle them. So there is landfills that are releasing the methane, and they have created the super challenges of the 21st century, we can say. The climate change, energy supply, health and diseases, and a sustainable environment. So the world climate change is intensely being discussed by various sectors as well as the stakeholders, because the life forms on earth is sustained through the proper climate as well as the further activities. So the two core issues of global climate change we can see is the rise in atmospheric carbon dioxide concentration and the temperature. So the warming of temperature also warms up the soil. So these changes in the temperature of soil will influence the soil microbial pattern. So once it's affected, the soil carbon and the nitrogen dynamics are also getting changed. And this is considerably studied under research at a larger scale, how to manage because the most of the carbon dioxide that are released into the atmosphere need to be sequestered and fixed by the plants. And the largest sequestering agent we know, the oceanic system. So in the oceanic system also the organisms are getting affected due to the climate warming. So there is a disbalance that have occurred. So this warming and the dried soil condition have increased the ammonia oxidizing bacteria and reduction in the abundance of ammonia oxidizing RK and denitrification bacteria. So this is affecting the nitrogen turnover in the agro ecosystem. So the extreme weather events of droughts or the heavy precipitation that leads to the flood is more frequent nowadays in different parts of the world. And this is affecting the agricultural ecosystems. And in addition to this, there are pathogens that are arriving. So due to the changes in the agricultural pattern as well as the climatic pattern have forced us to change the season, or the variation is there in the soil. This is leading to the change in the pathogens. The pathogen cycle is also changing because the extreme weather condition is giving an opportunity for them to survive and suppressing the other beneficial organisms. So the management strategies are practiced to reduce the nitrogen losses, improve the nitrogen use efficiency, and mitigate the global climatic changes. So these all are from the researchers that's giving a new dimension to the nitrification process. There is a requirement for the understanding of the microbial ecology and soil-plant microbial interactions in a changing climatic scenario. So the climate change adaptation and mitigation. These two terminologies are heard at a larger scale. And the other terminology that we hear is the climate resilience. So here we have to practice these words into real life. So the terrestrial polar regions, they are experiencing drastic melting of glaciers, sea ice, and permafrost. So these changes will change the microbial pattern, and these microbes are getting an access to decompose the previously frozen organic matter, and they are liberating a large amount of carbon. And this carbon again is modifying the carbon cycle of the atmosphere. And also these increased microbial activities, even though to some extent they are beneficial, they are also releasing the carbon dioxide and methane into the environment or the atmosphere. That's again increasing the Earth's temperature. So the effects of climate change on soil, lakes, rivers, and agricultural systems. See, these are related with the warming temperature as well as the greenhouse gases, and how these distribution are occurring in nature, and what is the changes that are occurring in the precipitation pattern, and how we can get a modified system of agriculture, how to manage this to support the agricultural production. So the land use changes are second to fossil fuels in shaping the climate change, because the land use changes, we know the forest cover was changed to the agricultural field, and the agricultural fields are now getting into the urban buildings or the industrial regions. So this is bringing about a change in the plant cover and the biogeochemical cycles. So this is influencing the tropical region as well as the coastal habitats, and the urbanization and industrialization. This is giving the another reason for the floods, because the natural pathway the water used to traverse is becoming interrupted by the human constructions. So that leads to the floods in the city or the townships, and also leads to larger disasters, we can say. So the conventional crop production, food production, where soil was tilled, results in the erosion and pollution runoff. So, and the changes in the precipitation pattern, temperature, and the surface. So these all are affecting the ecosystem carbon balance. And addition of nitrogen fertilizers to promote the crop growth. This also has added to the global nitrogen cycle. So slowly coming to the biodiversity part. So this is the driving force behind any healthy ecosystem. And a change in the pattern of temperature or variation in the land use pattern is also modifying the biodiversity. So it's providing many services in the ecosystem, a slight decrease in the species population, and it's negatively affecting the ecosystem's stability and maintenance. So the growing events of land use, land management for industrial and agricultural activities are modifying the terrestrial as well as the aquatic biodiversity. This results in the increase in temperature, decrease in rainfall, change in the seasonal variation, then increasing the frequency of cyclones, heat waves, and intensified wet or dry seasons. So this variation affects the whole of biodiversity. So I have given a concentration to the Indian agricultural scenario. So we know that the population is increasing at a larger scale, and as the population increases, there is a demand for the food production as well, along with others, because the food production makes the basic amenity for all. And most of the celebrations that we have in our system of Indian culture, we know they are all related, we can relate them to the agricultural season, either the harvesting festival or the sowing festival. So these were the natural or the pattern, the indigenous culture that we used to follow. But with the industrialization and advancement, there were changes that had occurred. And nowadays, due to the changes in the season, we are unable to utilize the same pattern for the agriculture. So the India is accounting for only 2.4% of the global land. So the land holding size is highly decreased. So we have the marginal and small farmers at the present time at a larger basis. So the agricultural census of the last agricultural census, we can say, they have shown that the agricultural production is increased. 49% of it is rice, then to the wheat, coarse cereals and pulses, then comes the fruits and vegetable production. We know that the Green Revolution have promoted the production of rice and wheat to manage the famine occurred at the time of. So now there is a need to think for the sustainable food production. So India was one of the first developing countries to prioritize the food security as a policy. So the self-sufficiency was achieved in 1970s through Green Revolution, where there was introduction of food crops with the hybridization as well as the fertilizer applications. But these have resulted into drastic changes in the agricultural pattern that not only affected the water sources and the soil fertility, but also had an impact on the climate. So it's critical that we need to create a suitable agricultural reforms. And if this is added with the incentive system for farmers, there will be adoption rate of sustainable practices at a higher rate. Because when they suddenly change from the traditional or the conventional pattern of agriculture of using agrochemicals to a sudden change into another sustainable practice, which includes or highly excludes the utility of chemical fertilizers, it will affect their economy. So there is a need to manage these through various policies, and solutions are to be brought. So the 26th session of the Conference of Parties at the UNFCCC, they have sparked many conversations. So here they have planning how we can adopt the mitigation pattern in different countries. We know that all over the globe, if we take, we are in the developing countries or the developed countries or the least developing countries. So we have to design the pattern according to the specific countries and how they can. So the agriculture is one of the most vulnerable sector to the climate or the weather pattern changes, because the largest food producers are about 68%, and almost all the population, either directly or indirectly engaged in agriculture, will be affected if there is a change in the agriculture. So there is a modification. We can say that the GDP has gone high from 1950s to the 2020, but these have also impacts on the. Ultimately, we can say that there is influence on the environment. So This is making India vulnerable to climate change. So the Economic Survey of India in 2017 have shown that the country is losing almost 9 to 10 billion US dollars annually due to the extreme weather conditions. That's either resulting in the crop losses or due to the pattern that is not suitable for the storage of grains. So there is a key challenge that is towards the food security and the livelihood. So the other resource-based enterprises that's related to the agriculture, to the horticulture, livestock, fishery, poultry, and the industries that are related with the agriculture. These all will be affected if there is a pattern of loss in the agriculture due to the climate change. So the United Nations Sustainable Development Goals that have uh, highlighted the zero hunger, nutrition and climate action. So the food production must double to by 2050 so that it will match the population and the economy of any country. So the assessment have also termed code red for humanity because as we know that the red list is for the endangered or the extinct species. So this is a code red for humanity where we are reaching to a point of no return because we are continuously exploiting the nature without considering for a future benefit. So those changes will ultimately lead to a condition that's difficult. So a more chaotic monsoon will have a grave bearing on the Indian agriculture as well as on the food production. So the rainfall also has sparked over the other crops. That's economically important crops. That's giving you the fibers or the oil crops because we know that even cottonseed oil is extracted nowadays and utilized for a edible oil. So all these are to be of concern because considering the climatic pattern, there is a modification that is made. So you can see that even the German researchers have compared the Indian monsoon with more than 30 state-of-the-art climate models from all over around the world, and they have issued this IPCC warning. So the economic cost of the climate emergency, it's singled out in 2020 report by Oxford Economics because the GDP could fall 90% by the end of this century if the current policies are not improved. The crop failures and the infestation are becoming more. And this is ultimately leading to the farmers' suicides. So to manage so many aspects of society as well as environment, there is a need to think over the next level. So 14% of the population is still undernourished in India. So this was a report of food security and nutrition in the world 2020. And also the Global Hunger Index 2020 has placed India at the 96th position among all the 107. So the achieving zero hunger is a motive that need to be achieved. So one of the critical challenges that we have is the climate change and its impact in the form of extreme weather events. The changes or increase in the temperature is going to affect the crop yield as well. So the high temperature may reduce the crop duration, permit the changes in the photosynthesis, and the crop respiration rates are getting escalated due to the changes in the increased temperature, and also the pest population. So the plant cannot survive through so many stresses, the biotic as well as the abiotic stress that they are facing through. And these cannot be managed by the agrochemicals or the genetic engineering that are practiced in the present condition. So this is going to affect the crop water as well as the soil because the water availability, changes in the intensity or frequencies, the population of microbes in the soil the organic matter reduction. So all these depletions are leading to a reduction in the productivity. So the economic survey has pointed out that the climate change might be reducing annual agriculture income in the range of 15% to 18% and up to 20 to 25% for unirrigated areas. So these situations will create the food shortages, nutrition deficiencies in humans, and this will be leading to the vulnerability of health. So considering different states of India, we can say from Himalayas to the coastal regions, the south countries need to be ready to fight against the impact of global warming. It's still assumed by the South Asian areas may face a warming effect up to 2 to 6 degrees in the 21st century. And the concentration of carbon dioxide are increasing or escalating. So various states of India are facing the dryness and they are prone to drought. So last seasons we can say there are so many people they are getting affected by the floods or due to the drought. And the worst heat episodes were also observed in Kerala. Because most of the Kerala's production, we can say the rice bowl is there. The super cyclone of Odisha, they took a change in the coastal region, and similarly the cyclone in Andhra. And all these changes are occurring due to the modification that have occurred in the pattern of biodiversity. Because mostly in the coastal zones there were mangrove plants that were supporting the areas or protecting them from the higher waves. But presently they are all getting adversely affected, and this is revealing. And the intense heat wave is experienced in India and the adjacent countries. And the highest temperature we can say it's crossing 50 degrees. And due to the reduced supply of water, there is increase in the salinity of the irrigated soils. So the 4 Intergovernmental Panel on Climate Change, it's releasing will be releasing its report on the changing monsoon, rising sea levels, the heat waves, the storms, flooding, and glacier melts. So these how they are going to risk the agriculture, and this will ultimately lead to a resource-hungry condition across the globe. So the Indian Council of Agricultural Research Farming is assuming consuming up to 30% more water due to high evaporative demand and crop duration due to forced mature. So this is occurring in states of Andhra, Punjab and Rajasthan. So similarly, the apple orchards in Himachal are shifting to higher altitudes for lack of solution in cold weather. Because the cold weather is most required for them to grow, flower and fruits. And the increasing population and demand for increased food production, India has to work more towards the emission of carbon dioxide and also to the alteration or the modification to the bioenergy system that can help. So most of the agricultural greenhouse gas emissions, we can say, are due to the agrochemicals and the agricultural inputs, the water, fertilizers, pesticides, and the machinery or the disturbance. So this sector is also partly responsible for the climate change. So the cultivation practices are completely based on the climatic patterns, because still till date we have the rain-fed system of agriculture that's practiced in different parts due to the lack of the river water supplies or the precipitation changes. So once there is a change in the precipitation pattern, this will induce the moisture stress in the soil, and moisture stress is leading to the sunburn and cracking symptoms in the fruits, and this will lead to loss of agriculture. And the ozone concentration is also reaching more than 50 parts per billion a day. So this is also leading a change in the influencing the crops especially, because the increased ozone concentration will influence the physiology of plants, and the changes in the physiology of plants will affect the uh, productivity. And also the animals body physiology is also getting altered due to the temperature, because there is a change in the heart rate and the blood flow and the body temperature, because these dairy breeds that we. Yes, hello, are you saying something? Is someone saying something? I don't hear.
No, ma'am.
Okay, thank you. So the dairy industry or the poultry industry are also going to be influenced due to these increasing changes of climate change and the fishery, because all these are dependent on the temperature as well as the water requirements. So the Government of India, Ministry of Agriculture and the Farmers Welfare and Indian Council, they are taking many policies and implementing many activities at different levels. To understand the impact of climate change on Indian agriculture and to develop the strategies, the Union Agricultural Ministry have formed the National Innovations on Climate Resilient Agriculture. So the study by this NICRA have shown that it's influencing the crops, livestock and fisheries in India. So, as we have already discussed in different regions, the temperature variation and the productivity, it's influencing the crops as well as the animals and also modifying the microbes we know. So the National Dairy Research Institute, Karnal, have found that the heat stress is adversely affecting the fertility of the cows and buffaloes. And this need to be considered, because we need, all of us, we know that without milk and milk products, or there is a difficulty, because in India there is a system of vegetarian food system. So we need to give more attention to this part as well. The ocean warming, this is also wiping out some of the commonly eaten fish, and there is also movement of fish, all the other aquatic organisms from the one region to the other due to these temperature patterns. Many a times we see that fishermen are getting different species of fish. That's due to the movement from one region to the other. So there is a need to aggressive adoption of sustainable farming solutions. These will address the food security as well as the ecological imbalance. Because majorly the ecological imbalance will lead to the changes that we need to address. So these programs are focusing on agroforestry, rainfed areas, water and soil health management, climate impacts and adaptations patterns. These are coming under the National Mission for Sustainable Agriculture. There are different programs, that is the Integrated Watershed Management Program, Krishi Sinchai Yojana. All these are on the micro irrigation, because how we can do the rainwater harvesting and utilize it for the future. So the studies conducted by the Council on Energy, Environment and Water in 2021 found that the sustainable agriculture is far from mainstream in India, because the changing to the sustainable agriculture means it needs to change from the agrochemicals to a source that is purely depending on the organic system. It will take time to adapt to this, because it needs time to get the soil fertility changed. So the research found that the 5 sustainable agricultural practices like crop rotation, agroforestry, rainwater harvesting, mulching and precision, this can be tried, and this can be only beyond 5% of the country's net zone area. And most of the farmers couldn't afford to try these alternative methods. To try these alternative methods means they have to uh, have a compromise on their economy. And these most of the marginal and smallholder farmers, it's difficult for them to adopt these. So the Ministry of Agriculture and Farmers Welfare, if they can have increased budget allocation towards this national mission, then they can have a thought for this. Because the higher temperature are cutting the productivity of workers. Because the hotter the day, lesser the ability to work under a hot scorching sun. So the outdoor workers, especially in the agricultural region, it's difficult. And the other risks are also there. In addition to this, the textile industry is also associated with agriculture. And these textile industries are also getting affected due to the increasing temperatures. So the study at University of Michigan's School of Business have shown that the garment factories are getting reduced work efficiency due to the increased temperature. And also these unpredictable weather and natural crises are leading to various patterns, that is a drought, pandemic, cyclones, heavy rains or floods. And this is disrupting the traditional agricultural calendar that we have seen, that our system of calendars and the celebrations or the festivals are associated with agriculture. So there is a modification that's occurring due to this. And this is also leading to a lower productivity due to the changes in the growth cycles. So there is a need for India to rejuvenate to at least 30 million hectares of the barren land to reverse to land degradation first. But this requires a large inputs, energy, efficiency, as well as the human input. And also the artificial intelligence system can be used here. That's being developed by the agricultural information sectors. So the precision agriculture can leverage the data analytics to maximize the efficiency of water, fertilizer and pesticides. So the efficient utilization of these resources can help to improve the field planning, soil fertility, monitoring and protection of crops, and also to reduce the biotic and abiotic stress on the plant. So the sustainable practices, as we have already discussed. So there is a need for replenishing the soil as well as the water resources. So moving our current agricultural production systems from an input intensive regime to low external input system, because large input of agrochemical is influencing the productivity. If we can reduce the agrochemical input and ability manage the organic system of or the organic matter of the soil, then it can be done. So the governmental organizations in different parts are working on it. So the zero budget natural farming, that's a concept introduced in Andhra Pradesh. And this is eliminating the chemical fertilizers and pesticides, and they are able to achieve to some extent. So in these approaches, the focus is on natural concoctions, inoculums and decoctions that are prepared from the natural ingredients. And in other case of Maharashtra, we can say that lower the use of water. That means how to conserve the soil moisture and promote the utilization of biofertilizers and biopesticides. But all these requires a patience of the farmers as well as the country. Promoting the organic farming system. So the union government schemes are there towards the organic and natural farming. That's towards the Rashtriya Krishi Vikas Yojana and the mission for integrated development of horticulture. So for examples, the Chhattisgarh have took the initiative to promote rural livelihoods along with organic farming. Then Godhan Nyay scheme is launched at Chhattisgarh. So here these farmers and the cattle ranchers, they are promoting the production of compost and utilizing it for improving the soil health. And in other case, Odisha has introduced the organic farming policy in 2018. So the Sri Sri University is the stakeholder, we can say. They are developing models in 30 different villages. They are developing these models to reduce the utility of agrochemicals, and in place they can produce a climate resilient agro ecosystem. So also they are planning, instead of the rice, wheat, they are also managing to promote the millets. And Uttarakhand is another example of Indian state. So here the organic farming policy was introduced in 2000. So this is the nodal state agency to promote the organic farming. So the Organic Agricultural Act 2019 was passed by Uttarakhand, making 10 of its blocks completely organic. So they will be having the pure policy of utilizing only the natural. Coming to the natural farming, Indian system, we can say the Indian states can step up to natural farming adoption. So this is led by the farmers and civil society from different states of India, where the chemical-free agriculture is being practiced. So the Niti Aayog calls it as natural farming because the words gives more palatable form. So the term natural farming is mostly used by the farmers, and the inputs are to rejuvenate the local ecosystems. So these are a few initiatives that are taken by the Government of India to promote the organic farming, so that there can be a climate resilient agriculture and also the sustainability can be achieved. So Dr. M.S. Swaminathan, who initiated the Green Revolution, have given a view towards the Evergreen Revolution in 2017. So this is to take pathways. He initiated it at the same place, the Agricultural University of Punjab, where he initiated the Green Revolution. So according to him, that there are different dimensions that we need to think about when we talk about the agriculture. It's not just the productivity, but also the employment opportunities, the sectors that are presently connected, the marketing linkages also. So considering all these, India has a rich traditional and wealth of indigenous knowledge that can be utilized or revived to the sustainable agriculture. Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Now the session is open for any queries and discussion. Participants, if you have any questions, you can put it in the chat box or unmute yourself.
Okay, thank you, Nisha. And I can see Manimekhalai, ma'am. Ma'am, unmute your mic, please, ma'am.
Thank you, ma'am. Very
Thank you.
informative session.
Thank you. Thank you
And
so much
also
for
we
giving
thank
the
our-
opportunity. I'll be connected as Dr. Gayatri has
Yes,
requested.
ma'am. Yes, ma'am.
Sure, ma'am. Sure, ma'am. Thank you, ma'am.
That will join now, ma'am.
Kiran?
Yes, ma'am. Yes, ma'am. Now I can see you, ma'am.
Okay. On the behalf of Avinashilingam University, I am very, very grateful to you for joining.
Thank you
And
so
it was
much, ma'am.
very
Thank you so
informative,
much.
and I'm also happy about it. All the speech was very interesting, and it will be useful for all the scholars and the participants.
Thank you so much, ma'am. Thank you so much.
Thank you, ma'am. Your presentation about climate change, sustainable practices of agriculture, and organic farming was really inspiring. And thank you, sir, so much for taking time. Thank you, ma'am.
Thank you so much.
Warm afternoon to one and all gathered. It's time to start our valedictory ceremony. I'm happy in welcoming you all to the valedictory ceremony of UGC Stride-sponsored 6 days training program on environmental sustainability and research ethics. One song can change a moment. One idea can change a world. One step can start a journey, but a prayer can change the impossible. I now request Ms. Bhavana from 2nd PG Biochemistry to make the session delightful with her hymn.
Bhavna?
Yes, ma'am.
Just wait a minute. Sir will join now. Okay.
Okay.
The last session speaker, he'll be doing the valedictory address. So just wait for a few minutes.
Okay, ma'am.
Then you can start with your prayer song.
Okay, ma'am. Okay.
Thank you. So welcome, sir. Good afternoon, sir. Sir, join பண்ணிட்டாருமா?
Okay.
Good afternoon, sir.
Sir, join பண்ணிட்டாரு.
Good afternoon to everyone.
Good afternoon, sir.
Good afternoon to everyone.
Welcome, sir.
Good afternoon, sir. I am Dr. Manimekale from Department of Zoology.
Good afternoon, madam. Thank you very much.
உங்க ஆப்போசிட்ல தான் சார் நான் ஸ்டடிட் மை எம்ஃபில்.
என்ன மேடம்?
கேம்பஸ் சிஎஸ்.
Okay.
ஆமாங்க சார்.
Very good, madam.
Good afternoon, sir.
Good afternoon, madam.
Good afternoon. On behalf of Avinashilingam University, I welcome you, sir.
Thank you, madam.
Now I request Ms. Bhavna from 2nd PG Biochemistry to sing a prayer song.
चरणु सिद्धि विनायक चरणु विद्या प्रदायक चरणु सिद्धि विनायक चरणु पार्वती तनय मूर्ति चरणु मूषिक वाहना चरणु पार्वती तनय मूर्ति चरणु मूषिक वाहना चरणु सिद्धि विनायक चरणु विद्या प्रदायक चरणु सिद्धि विनायक. Thank you.
Thank you, Ms. Bhavna. I now request Dr. G. Maheshwari, Assistant Professor, Department of Botany, to welcome the gathering.
Wonderful morning is over. Here is afternoon wish for you to spend an incredible midday. With the blessings of God Almighty, our founder Avinashilingam Ayya avargal, world famed nutritionist Rajama P. Devadas Amma avargal, and meticulous support from our Honorable Chancellor Professor S.P. Tyagarajan, Vice Chancellor Dr. Bharati Harishankar, Registrar Dr. S. Kausalya, the Department of Zoology, Botany, Biochemistry, Biotechnology and Bioinformatics and Physician Assistant organized 6 days online training program on environmental sustainability and research ethics for Indian faculty and research scholars of Avinashilingam University, Coimbatore. It gives me an immense pleasure in welcoming all of you for the valedictory meeting of this event. This training program has been organized to discuss the various issues to sustain our environment and the research ethics that should be maintained while doing any activity. I feel honored to welcome Dr. P. Palani, Professor, Department of Botany, University of Madras, Chennai, to give valedictory address. Next, I feel proud to welcome UGC Stride Project Coordinator, Dr. K. Manimoli, Professor, Department of Resource Management, Controller of Examination. Welcome you, ma'am. I am very happy to welcome our Dean, School of Biosciences, Dr. A. Vijay Lakshmi, for the valedictory meeting. It's my duty to invite our conveners, Dr. Anita Subhash, Professor and Head, Department of Biochemistry, Biotechnology and Bioinformatics, Dr. K. Manimegalai, Professor and Head, Department of Zoology, Dr. A. Uma Devi, Assistant Professor, Department of Physician Assistant. I am very delighted to welcome the organizing secretaries of this program, Dr. P. Kartiga, Department of Zoology, Dr. M. K. Nisha, Dr. V. Gayatri, Department of Botany, Dr. S. T. Yamuna and Dr. C. C. S. Vasundhara, Department of Biochemistry, Biotechnology and Bioinformatics, and Mrs. Ashwini, Department of Physician Assistant. It's indeed a matter of great honor to welcome the faculty members and research scholars all over India for participating in this fruitful training program. I am sure that you are all were enriched with knowledge and interacted with highly qualified eminent professionals in various fields, and I expect that these discussions were beneficial for your future venture. Once again, I welcome you all for the valedictory session to make this training program as a memorable and successful event. Thank you.
Thank you, ma'am. Next, I invite Dr. K. Manimegalai, Professor and Head of the Department of Zoology, to present the training program report.
Good afternoon to one and all present here. As a part of the quality mandate, the University Grants Commission has introduced Scheme for Transdisciplinary Research for India's Developing Economy, a new scheme to promote quality research by faculty and students. The specific focus of the scheme is to support high-impact research in trust areas of humanities and human sciences, including arts, Indian languages, social, cultural, environmental, economic, political context, and knowledge system through networking of experts from institutions and organizations across India. This would promote new knowledge, inculcate innovative and cognitive thinking, and improve doctoral research quality. The main objective of the scheme is to develop a research culture and interest among young faculty, scholars, and students to pursue careers and undertake high-quality research in transdisciplinary areas that augment and expand the research capacity. Keeping this objective, we have organized for 6 days online training program on environmental sustainability and research ethics from 21/3/2022, 26/3/2022 with 24 speakers from all over India, expertise in the topics of quality research and environmental sustainability. 421 participants have registered for the event, and 229 participants from Tamil Nadu, Kerala, Karnataka, Telangana, Maharashtra, Uttar Pradesh, Uttarakhand, Assam, West Bengal, Odisha, Gujarat, Rajasthan, Jammu and Kashmir, Assam, Puducherry, Lakshadweep, and Arunachal Pradesh have attended the training program. The link for Google Meet and YouTube live streaming link was sent prior to all the participants. The inauguration of 6 days online training program on environmental sustainability and research ethics commenced at 10 a.m. with a prayer song by Miss Sweta, Department of Music, Avinashilingam Institute for Home Science and Higher Education for Women, Coimbatore. The master of ceremony for the day one was done by Miss M. Sandanambigai, research scholar, Department of Zoology, and Miss C.V. Prithika of 3rd BSc Zoology of our institute. Dr. Anita Subhas, Professor and Head, Department of Biochemistry, welcomed the gathering. Dr. A. Vijayalakshmi, Professor and Head, Department of Botany, Dean School of Biosciences of our institute, greeted the participants. The inaugural address was given by Dr. P. Ponmurugan, Associate Professor, Department of Botany, Bharatiyar University, Coimbatore, on the topic Patent Drafting and Filing Procedures in India and Abroad. He emphasized on the importance of filing patents and the various steps about filing it. He also gave an overview on trademarks, copyrights, and trade secrets. The formal vote of thanks was proposed by Dr. K. Manimekalai, Professor and Head, Department of Zoology of our institute. Day 1, 4 resource persons addressed on 4 different topics. Morning sessions were by Dr. P. Ponmurugan, who explicated on patent drafting and filing procedure in India and abroad, followed by Dr. Sudha Ramalingam. We expounded on basics of research ethics. The session commenced after a lunch break for 2 sessions by Dr. Subhashini K. Sripati and Mrs. G. Geeta, who delivered a talk on patents and copyright and plagiarism checking and research, respectively. Each session had 1 to 15 minutes interval for discussion where the resource persons addressed and the queries and clarified the doubts of the participants. Day 2, morning session was presented by Dr. Nitin Keshav Srinivasan, Medical Director and Consultant, Madurai Eye Hospital, who explicated on diabetics retinopathy, do's and don'ts, followed by Dr. M. Satyabama, Professor, Department of Botany, Bharathidasan University. We expounded on nanotechnology for enhanced crop productivity. The first lunch session was presented by Dr. Swet Joshi, Scientist B, National Institute of Occupational Health, on reference management and scientific publications, followed by Dr. Balasubramanian, visiting professor on the statistical methods in research. Each session had 15 minutes interval for discussion where the resource persons clarified the doubts of the participants. Day 3, in the forenoon, the first lecture was delivered by Professor Parimal Alagar from Bharatiyar University, Coimbatore, followed by Professor S. Acharya from Institute of Bioinformatics and Applied Biotechnology, Bengaluru, who discussed about research ethics. An open discussion and debate, the post-lunch sessions were delivered by Dr. S. Yamani Sudhalakshmi from University of Madras and Dr. M.S. Vasanta Lakshmi from All India Institute of Speech and Hearing, Mysore. At the end of each session, the research persons actively interacted with the participants and clarified their queries. Day 4 in the morning, the first session was delivered by Dr. Devrata Das, Associate Professor and Head, Department of Botany, Government General Degree College, on the ecological assessment of health and environment through vegetation. The second session was given on fungal biofertilizers and its application in agriculture and forestry by Pampi Ghosh, Assistant Professor of Botany, Shaiva Bharati Mahavidyalaya. In the afternoon, the third session was handled by Dr. B.A. Daniel, Director Research and Education, Secretary Zoo Outreach Organization, Coimbatore, on if sustainable utilization is needed. The last session of the day was presented by Dr. S. Vidyalakshmi, consultants, obstetricians, gynecology and fertility specialists, Excella Gynocare Women's Specialty Center, Saravanapatti, Coimbatore, on cervical cancer. Day 5, the first session was handled by Dr. A. Arumugam, Professor, Department of Botany, Alagappa University, Karaikudi, on gene in environment. The second session was by Dr. S. Muralidharan, Salim Ali Center for Ornithology and Nature History,
Manakatti, Coimbatore, who delivered a speech on sustainable use of chemical pesticides and the ill effects on the environment. The third session was delivered by Dr. Thangarasu on statistics for data science and business analysis. The last session was delivered by Dr. Kannan Warrior on sacred truths and their incomparables. Day 6, in the forenoon, the first lecture was delivered by Dr. Sheila Sasikumar, Director Clinical Research, SS Healthcare Chennai, on the topic Air Pollution, Climate Change and Human Health, followed by Dr. K. Jayabharati, Professor in the Department of Pediatrics, PPG College of Nursing, Coimbatore, who emphasized research design. The first length sessions were delivered by Dr. R. Hiranmay Yadav, Professor, School of Environment and Sustainable Development, Central University of Gujarat, who discussed about climate change and agriculture. The feedback link was posted at the end of the day, and the responses were received from the participants for all the days. Thank you.
Thank you, ma'am, for the detailed report. Better than 1,000 days of diligent study is one day with a great teacher. I now request Ms. Dhanushree to introduce such a person, our guest, Dr. Parani, Professor, Center of Advanced Studies in Botany, University of Madras, to the gathering.
It's my privilege to introduce our guest, Dr. Palani, Professor, Center of Advanced Studies in Botany, University of Madras, Chennai. Dr. Palani had completed his doctoral research from Center for Advanced Studies in Botany, University of Madras, Chennai in 2002, followed by postdoctoral research fellowship in Department of Microbiology and Immunology, Texas Tech University Health Sciences Center from 2001 to 2006. He has 24 years of research experience at national and international laboratories. Dr. Palani had published about 66 research papers in reputed peer-reviewed national and international journals. He had successfully completed many research projects funded by UGC, DST, SERB, and MHRD for about 100 lakhs. Under his guidance, 3 researchers had completed postdoctoral research and 11 students had successfully completed PhD and 27 students had completed MPhil. He had worked as a visiting faculty at the Department of Microbiology and Immunology, Texas Tech University Health Sciences Center, Lubbock, Texas, USA. He had received several awards and honors, namely the Best Research Paper Presentation Award by the School of Medicine, Texas Tech University Health Sciences Center, USA in 2004. The Dr. Kalyanasundaram Memorial Award for the Best PhD Thesis by the University of Madras in 2003. Best Researcher Award by the University of Madras for the academic year 2018 to 2019 in September 2018. Best Researcher Award by the University of Madras for the academic year 2019 to 2020 in September 2019. We welcome you, sir.
Thank you.
Participants are kindly requested to mute themselves.
Okay, so is there any other formalities or shall I start my lecture?
Sir, you can
No,
start.
sir, you can proceed.
Okay, good, okay. Thank you very much. It gives me immense pleasure to be back with the Avinashilingam University, Coimbatore, to deliver yet another valedictory lecture. And many of the faculty members of the Department of Botany knew that I have already delivered a valedictory lecture earlier. And so I'm extremely happy to deliver another valedictory lecture of an online training program on environmental sustainability and research ethics. And I have just listened to the report presented by the HOD of Zoology Department, and I observed that many of the faculty members have delivered their lectures, and hopefully the participants would have absorbed all the contents delivered during the lectures. And before I could start my lecture, I would like to thank the faculty members of Department of Botany and Department of Zoology for putting all your efforts in organizing this online training program. And I knew how much effort is needed for organizing such a meeting or training program. So with this brief note, I have great pleasure and I'm happy to present a small lecture on a topic which is very familiar with all your participants. And let me share the presentation slides. Please, one of you tell me whether you are able to see the slide
Yes.
or not.
Yes, sir, it's visible, sir.
Okay. So this is actually the topic I would like to talk about today as part of the valedictory lecture of the training program. And the title is Recent Advancements in Proteomic Analysis. And here is my address. If any of the participants would like to ask a question or clarifications, either during the lecture time or even after the lecture gets over, you are very welcome to contact me through the email which has been given as palani7@unam.ac.in. And I begin my lecture by introducing the electrophoretic analysis of proteins. Of course, I don't have to explain what a protein is. And every one of you might be knowing what is a protein is. And what I'm going to focus now is, I will be focusing my attention on how the proteins are analyzed using different types of electrophoretic techniques, which will be highly useful for all the participants, irrespective of their disciplines. So some of the participants may be from a discipline of botany or zoology or chemistry or environmental microbiology, biotechnology. But the lecture I will be delivering today will be highly useful to all the discipline and the participants, all the participants of this training program. So when you want to analyze a protein, and we use two different types of electrophoretic analysis. The first one is native page, and the second one is SDS page. In fact, the electrophoretic analysis of protein was discovered very first with native page. So this is the first technique which was discovered, and later on some kind of developments were introduced in the native page itself, and then the modified electrophoretic analysis came to be known as SDS page. And I would like to draw the attention of all the participants. The preparation of gel in the preparation of the samples and the electrophoretic separation of proteins using both the native page as well as the SDS page are almost similar. But we'll have to see the specific applications of these two techniques. Native page. I will talk about the native page first. And before I could explain about native page separation of protein, and you participants need to know the various structural levels of proteins. There are primary structures. The primary structure is nothing but amino acids in a linear chain. So that is called the primary structure of protein. And the secondary structures, the next level of a structure is the secondary structures. And the secondary structure can be divided into two different types, alpha helix and then beta pleated sheets. Then comes the tertiary structures. And the fourth level of structure is called the quaternary structures. Except the primary structure, the remaining three levels of proteins, secondary structure, tertiary, and the quaternary structures. And in these secondary, tertiary, quaternary structures of protein, several forces like covalent bonds, van der Waals force, ionic interactions, and other things, hydrogen bonds, and all of them participate in providing a specific shape to the proteins. And we call the proteins as native proteins. So this will be the structure of a native protein. And if you are going to electrophorese a protein with its native conformation on an electrophoretic system, then the system is going to be called as native page. Remember, the proteins have four different levels of structures: primary, secondary, tertiary, and quaternary structures. And we are not going to destroy these secondary structures before analyzing them on the polyacrylamide gel electrophoresis. So those electrophoretic techniques which uses proteins without changing its native conformation are called native page. And in the second case, it's called the SDS page. What we do here, so this is the secondary structure of a protein. Secondary structure, tertiary, and quaternary structure of protein will be like this. But what we do now, we treat that protein with a denaturing agent called SDS. And SDS will denature the protein, and after denaturation, the protein will become linear in shape. So previously, before the treatment of SDS, the protein was in a native conformation, which will have a secondary, tertiary, and quaternary structures. But after the treatment of denaturing agents, and the most commonly used denaturing agent is sodium 2-decyl sulfate. In short, it is called SDS. And after the treatment of SDS, the protein will be denatured, and the denatured protein will appear as a linear shaped protein. So this is the basic difference between native page and SDS page. And I began my lecture that the native page electrophoretic technique was the first one which was discovered. And this technique is not sufficient enough to analyze various different types of proteins. In a given cell, consider this is as a bacterial cell or a plant cell, and the cell will have hundreds and thousands of proteins. And some of them will be positively charged, and other group of proteins will be negatively charged, and still other proteins will be neutrally charged. So there will be positively charged protein, negatively charged protein, and neutrally charged protein also will be there. There are three major group of proteins available in a given cell, whether it is a bacterial cell or plant cell or animal cell or human cell. So this is the case. And so what you have to do before electrophoresing the proteins isolated from your cell? So this is the cell. Consider this is a plant cell or animal cell, and you are going to extract the proteins. And the extract will have three different population of proteins: positively charged protein, negatively charged protein, and neutrally charged protein. And the major disadvantage available with the native page is the native page can separate only a specific charged proteins. Either you can separate the positively charged protein or negatively charged protein, but we cannot separate both positively charged, negatively charged, and neutrally charged protein together using a native page. So that is a disadvantage available with the native page. So at a time, you can either separate positively charged protein or negatively charged protein, but not both at the same time. So if you can analyze only a positively charged protein at a time, for example, if you are going to analyze positively charged protein alone, and you will be missing the information about how many proteins are negatively charged and how many proteins are neutrally charged. So these two informations will be missing. But at the same time, if you want to analyze a negatively charged protein, then you will miss the information about positively charged protein and the neutrally charged protein. So therefore, a modification was necessitated, and a discovery of a new format of electrophoretic technique came into being. And that technique is called SDS page. In SDS page, so in a given sample, usually there will be three different proteins. So positively charged protein will be there, negatively charged protein will be there, and then neutrally charged protein also will be there. So here is the extract. Assume that you have got the extract from bacterial cell or plant cell or animal cell, depending upon your discipline. But we want to see all the proteins. I want to get all the proteins on a single electrophoretic technique. Therefore, after the discovery of SDS page, we were able to analyze all the 3 different proteins. And only a single modification was done from the native page to SDS page. In fact, the reagents used for preparation of the native page and the reagents used for preparation of the SDS page, all of them remain same. But only an additional component is introduced into the SDS page, that is called the SDS. Addition of SDS, when you prepare the gel, of course there are 2 different layers. I don't have to explain them. A gel will have 2 different layers, and the upper portion of the gel is called stacking gel, and the lower portion of the gel is called resolving gel. And when you prepare the resolving gel, both resolving gel and the stacking gel, SDS will be incorporated into the gel. And after the preparation of the gel, the gel will be connected to a tank, electrophoretic apparatus, and the lower tank and the upper tanks will be filled with the electrophoretic buffer. And the buffer also has to be incorporated with the SDS. So when you prepare the gel, the SDS has to be incorporated. Secondly, when you prepare the electrophoretic buffer also has to be incorporated with the SDS. And in the third step, after the preparation of gel, and after connecting the gel to an apparatus, the protein sample has to be loaded in the wells. And before loading these samples, the sample has to be treated with a buffer containing SDS and beta-mercaptoethanol. Of course, the sample buffer will have different components like tris buffer pH 6.8, glycerol, bromophenol blue, water, and sodium 2-decyl sulfate, sodium SDS, and beta-mercaptoethanol also will be there. And here is the protein mixture that you have isolated from a source organism. And the protein sample has to be mixed with a sample buffer containing SDS and beta-mercaptoethanol. And as I mentioned earlier, when you treat the protein sample with SDS, SDS will denature all the proteins, irrespective of whether it is positively charged protein or negatively charged protein or neutrally charged protein. The SDS will interact with all types of protein and will make them as a linear polymers. Not only that, the SDS also binds with the protein. And now, once you treat the protein sample with the SDS alone, 99% of the proteins will be denatured. Denature is nothing but removal of the secondary, tertiary, and quaternary structures. Technically speaking, removal of a secondary, tertiary, and quaternary structure is called denaturation. And you may ask a question, when we can denature the protein only with the treatment of the SDS alone, and what is the necessity to use beta-mercaptoethanol? So that question will be obvious in the minds of everyone. And I would like to tell you that the proteins, so here is a secondary structure, and there will be amino acid called cysteine. Cysteine amino acids will be there. The presence of cysteine amino acids in the protein will be at many different sites. So we have a cysteine amino acid in this particular place, and we have another cysteine amino acid in a different site. So between these two cysteine amino acids, there will be disulfide bond formation. And the disulfide bridge formation is a covalent formation. And even though we are treating the protein with the SDS cannot break the disulfide bridge between two cysteine molecule in a protein. Therefore, if you treat the protein with the beta-mercaptoethanol along with other components in the soluble buffer, the chemical bond between two cysteine, the covalent bond between two cysteine amino acids will be broken, and the protein will be completely released into a linear polymer. So this is the reason why we add sodium dudecyl sulfate and beta-mercaptoethanol in the soluble buffer used for SDS page. Okay, and now, so this is the developments that has taken place between native page and SDS page. And after that, I would like to also mention about the mechanism of polymerization, and many of the participants may not be aware of it. So for the benefit of all those participants who are not aware of this, the mechanism of polymerization, I would like to give you a brief information about how the polymerization of the gel occurs. And you know that the polyacrylamide gel is prepared with acrylamide monomer. And what we do, we have to create and we have to generate the free radicals. And the acrylamide polymerization actually requires the free radicals. And for the production of free radicals, we use a chemical called ammonium persulfate. And the specific characteristics of this particular chemical is, if you dissolve ammonium persulfate in water, ammonium persulfate will decompose immediately and will produce free radicals. So this is the natural process. And after the production of free radicals, and the acrylamide monomer, so this is the chemical structure of acrylamide monomer, one monomer of acrylamide. You don't have to worry about the chemical formula or chemical structure. Remember only the name. of the chemical. It's acrylamide. So this is the monomer of acrylamide. And one monomer of acrylamide will capture the free radical generated from the decomposition of ammonium persulfate. And then the ammonium persulfate will produce free radicals, and these free radicals will be captured by the monomers of acrylamide. And previously, the acrylamide monomer was in an inactive form. But now, after the capture of a free radical, and this monomer now will get activated. So the acrylamide monomer now is activated. And the activated monomer, in turn, will interact with another monomer of acrylamide. So this is the monomer which is not activated, the second monomer which is not activated. And here is the monomer which has already been activated by the free radicals. The activated free radical, the activated monomer of acrylamide will interact with the unactivated acrylamide monomer. And during this time of interaction, a chemical bond will be formed between two acrylamide monomers. And now, two monomers of acrylamides are joined. And then the second monomer now will get a free radical from the decomposition of ammonium persulfate. And this in turn will be activated. And the activated monomer will interact with another monomer of acrylamide. And a chemical bond will be formed. And the third monomer now will get the free radicals. And this in turn will interact with the fourth monomer of acrylamide. And finally, what we are going to get is a linear polymer of acrylamide, like this. And again, I stress, you don't have to worry about the chemical structure. Remember only the names. So here is acrylamide monomer. And the acrylamide monomer has been activated by the free radicals. The activated monomer will interact with other unactivated monomers, and they will be combined together in a long linear chain. So this is actually one chain of polymer, acrylamide polymer. And here is the another chain of polymer. And in reality, when you do polymerization by yourself, you are going to do the preparation of gel in a small volume like 10 ml or 15 ml. Then 15 ml of gel solution will have millions of acrylamide monomers. And all the millions of acrylamide monomer will receive the free radicals from the ammonium persulfate decomposition, and they will be activated. And after the activation, the activated monomer will interact with another monomer, and they will be joined together. Several thousands of monomers will join together and finally will form a long linear chain. And so in addition to the formation of long linear chain of acrylamide, so this is chain number 1 and this is chain number 2, and these two chains have to be cross-linked. And the cross-linking function will be done by another chemical called bisacrylamide. So, several thousands of linear polymer chains will be cross-linked by the bisacrylamide monomer. And before I could go further, I would like to give a little more information about the polymerization of acrylamide gel. Acrylamide polymerization can be accomplished with using a chemical, or with it can be also polymerized with a light. If you expose the gel solution to a light, after the addition of a chemical called riboflavin, And when you expose the riboflavin to sunlight, the riboflavin will decompose and will produce a free radical similar to the ammonium sulfate decomposition. In both the cases, the final product is the free radicals. And once the free radicals are produced, and they will be captured by acrylamide monomers and they will be activated. Activated monomers will interact with the other monomers and long linear chain will be formed, and these chains will be cross-linked by bisacrylamide. So like this, the polymerization process will occur. And ultimately what we are going to get, as many of you are aware, that ultimately we are going to get a semi-solid gel. And so you know that the gel is prepared between two glass plates, and the two glass plates will be separated by spacers. And the purpose of putting the spacer is to give uniform thickness to the gel inside. And now the spacers are put on either side and also at the bottom of the glass plates. And after aligning the other plates also, the margins of the glass plates will be sealed with either molten agar or grease. And after that, we'll have to prepare the reagent gel solutions. Don't go very deep into the details of the reagents. Remember only the names. So we need 3 molar tris buffer pH 8.8. We need 0.5 molar tris buffer pH 6.8. And we need another solution called acrylamide bisacrylamide solution. And the proportion of acrylamide to bisacrylamide is 30% of acrylamide and 0.8% of acrylamide. And you'll have to prepare 0.14% ammonium persulfate if you want to run native page. But 10% ammonium persulfate has to be prepared if one wants to run SDS page. And then we have to prepare the electrophoretic buffer. The electrophoretic buffer for native page will not have SDS in it. But the electrophoretic buffer, which has to be used for SDS page, has to be incorporated with the SDS. Similarly, here is the sample buffer. Sample buffer, as I told you before, the sample buffer naturally will have the tris buffer, 0.5 molar tris buffer, pH 6.8. This is the buffer. So this is the buffer from which you will have to take 1.25 ml of buffer, and then deionized water has to be added, glycerol has to be added, and then bromophenol will be the fourth component of the buffer. And this is the sample buffer which is used for native page. And if you want to use the sample buffer for SDS page, two other components have to be added. SDS has to be added, and then beta methoctanoethanol has to be added. So if you add these two components in the sample buffer which was prepared for native page, then this sample buffer now will be suitable for SDS page. So this is the basic difference between the sample buffer used for native page and SDS page. And all the reagents, whether it is a 0.5 molar tris buffer or 3 molar tris buffer or acrylamide bisacrylamide solution, ammonium persulfate, only the concentration percentage of ammonium persulfate will differ from native page to SDS page. For native page, it is 0.14%. And for SDS page, it is going to be 10%. And then electrophoretic buffer. The electrophoretic buffer for both native page and SDS page will have tris buffer along with the glycine. Tris and glycine will be the two components which will be commonly used for both native page and SDS page. And if this buffer is going to be used for SDS page, then you'll have to add SDS in it. So these are the difference between native page and SDS page. And now, if you want to prepare the resolving gel, you'll have to prepare resolving gel and stacking gel. And some people will be worrying about how much volume has to be taken, how much volume of acrylamide has to be taken, and how much volume of 3 molar tris buffer has to be taken, and how much volume of ammonium persulfate has to be taken. So, if one wants to prepare 10% of gel, you can prepare different percentage of gel depending upon your requirement. Some will require 15% gel, others will prepare 10% gel, and still others will prepare 7% gel, and the preparation of the percentage of gel will be decided by the what type of protein one is going to analyze. And now we are going to prepare 10% of gel, 10% of acrylamide gel, resolving gel. And to prepare 10% of resolving gel, you'll have to take 3.3 ml of 30% acrylamide and bisacrylamide solution, 1.24 ml of 3 molar tris buffer, pH is 8.8, and 0.5 ml of 0.14% ammonium sulfate. And then 4.9 ml of glass distilled water and TEMET will be 10 microliter. And the total volume will be 10 ml. So, we have to put all the reagents together, and then when you put all the reagents together, then the polymerization process will occur. And within 20 to 25 minutes, the polymerization will be complete. And once the resolving gel is prepared, then you'll have to prepare the stacking gel. And the stacking gel, these are the components. How much acrylamide has to be taken, how much tris buffer has to be taken, how much ammonium persulfate has to be taken, and how much volume of water has to be added. All the details are provided here. And similarly, what we have done before for the preparation of resolving gel, the stacking gel also will be prepared. And once you come to know that the resolving gel has already polymerized, then you can layer the stacking gel on top of it. And now you can, after the polymerization, the gel plate will be attached with an apparatus. And before attachment of the glass plates, along with the gel, polymerized gel in between, the spaces present at the bottom of the gel has to be removed. So why we have to remove the spaces? Because we have to establish the connectivity between the upper tank and the gel and the lower tank. So the buffer available in the upper well should have contact with the upper part of the gel. And the buffer which is put in the lower tank of the apparatus should have contact with the lower part of the gel. So that we can supply electricity. So that is the reason why you'll have to remove the spaces at the bottom of the gel. And once you fix the apparatus, once you fix the gel plates with the apparatus, and the tanks will be filled with the electrophoretic buffer, and power supply will be connected. So here is the positive electrode, and here is the negative electrode. The gel has already been fit in an electrophoretic apparatus, and the lower and upper tanks are filled with the electrophoretic buffer. And after filling the tanks, the sample has to be loaded. And before loading the sample has to be mixed with the sample buffer containing all the components that I have described before. The sample buffer will have tris buffer, glycerol, deionized water, bromophenol blue, and in the case of SDS page, the sample buffer will also have SDS and beta-mercaptoethanol. And after treating the sample with the sample buffer, the sample will be loaded into the wells like this. And after loading the sample, the electrophoretic apparatus will be connected to an electric source. Now, what I'm going to describe is the principle of separation of proteins. So this, many of the participants will not be aware of it. So here is the stacking gel, and this is the resolving gel. And now we have loaded our sample. Here is the wells, and we have loaded our sample here. Now the electricity will be turned on. And what is going to happen, as I told you before, when you treat the protein with the sample buffer containing SDS and beta-mercaptoethanol, and SDS will bind with all types of protein, whether it is positively charged protein or negatively charged protein or neutrally charged protein, all of them will be bound with the SDS. So therefore, the native charge of the protein will be masked by SDS. Now, after treating the sample with the SDS containing sample buffer, the sample is now loaded, and now we are going to supply the electricity. And what happens now, as soon as you turn on the electricity, and look at the pH of the separating gel. The pH of the separating gel is 6.9, while the pH of the resolving gel is 8.8. Okay, you'll have to remember that the sample buffer that we have prepared for SDS page contains a buffer, tris buffer. And that tris buffer has chloride ion. And also, it also has glycine in it. The electrophoretic buffer has glycine. We have prepared the electrophoretic buffer with the glycine. So therefore, the well in which we loaded our sample will have both chloride and glycine ions along with our protein. So there will be three components in the well. Our protein will be there, our protein mixture will be there, and glycine will be there, and chloride ions will be there. So these are the three components which will be present in the sample well, in addition to other components which do not play any major role in the separation of proteins, like the glycerol and other things. They do not play any role. But glycerol provides only one role, which gives density to the sample. Okay, so now the sample has been loaded, and now look at the pH. pH is 6.9. At that pH, the glycine, negatively charged glycine ions, originally the glycine anions will be negatively charged. But because of the change in the pH here is 6.9, and all the negatively charged glycine anions will be converted into zwitter ion. So when you say zwitter ion, an ion which has both positive and negative charge in equal proportion. So originally the glycine will exist in the negatively charged form, but because of the change in the pH in the separating gel, and the negatively charged anions will be converted immediately into zwitter ions. Therefore, when it becomes Zwitter ion, it cannot carry electricity. And the Zwitter ion will become immobile under pH 6.9. So I told you three different components: so protein, chloride anion, and glycine ion. And one of the ions, glycine anions, now has been converted into Zwitter ion, and they cannot conduct electricity because they don't have any charge on it. They are neutrally charged. When they are neutrally charged, they cannot conduct electricity. And now we have two other components. The sample. Our sample is completely converted into negatively charged proteins. After treating the protein with the SDS, all the proteins will be converted into negatively charged protein, irrespective of whether it is positively charged or negatively charged or neutrally charged protein earlier. And the third component will be chloride anion. Negatively charged chloride ion will be there. So now, soon after supplying the electricity, and most of the current that we supply to the gel will be conducted by the chloride anion and the protein. So now you focus your attention here. So the glycine, all the glycine anions will be converted into neutrally charged Zwitter ion. So therefore, the upper black one is the glycine anions. Since all the negatively charged glycine ions are converted into Zwitter ion, they cannot move and they will stay at the top of the gel. And now we have the two ionic components, and one ionic component is the chloride ion, negatively charged chloride ion, and the second ionic component is our protein. Both are negatively charged ionic components. So chloride ion will be moving first. So this is the chloride ion, followed by proteins, and followed by the neutrally charged Zwitter ion. And you also have to focus your attention that the percentage of stacking gel is always 5%. So when the percentage decreases, the pore sizes in the gel will be larger. While we prepare the resolving gel with the 10% gel, and the stacking gel is prepared with 5% acrylamide. So whenever you reduce the concentration of the acrylamide, the pore sizes, after the formation of gel, the gel will have minute pores in it. And the size of the pores will be larger if the percentage of acrylamide is less. But when you increase the percentage of acrylamide from 5% to 10%, the pore size also will correspondingly decrease. So, though the glycine Zwitter ion do not conduct any electricity, and because of the large pore size present in the stacking gel, they move passively. And now the order of movement is the chloride ion always will go first, followed by protein, and followed by glycine Zwitter ion. So these changes. So our protein has been left between chloride anion and glycine Zwitter ion. So it is now sandwiched between chloride and glycine ions. So these changes, okay, now compress the proteins. We have mixture of protein, and all the protein will have all different types of proteins. A larger molecular weight protein will be there, medium-sized protein will be there, and the low molecular weight protein will be there, and all of them will be compressed together and will stack. Not only compressing, but also will stack one above the other. Protein will be stacked one above the other like this. So that is why this gel layer is called stacking gel. And now we come to the resolving gel. So after a few minutes, all the three ionic fronts will reach the surface of the resolving gel. And now there is a change. There is a sudden change in the pH from 6.8 to 8.8. And as soon as the ionic front reaches the surface of the resolving gel, it encounters a change in the pH, 8.8. And because of the increased pH, previously the glycine was in its sweeter ionic form, and now will be converted into glycine anion. Because of the change in the pH, all these sweeter ions will be converted into negatively charged anions. So now, because of the conversion of the glycine sweeter ion, and when it is transformed into glycine anions, now the glycine anion also will conduct the electricity, and the movement of the glycine anion will be faster than the protein molecules. So therefore, when it reaches the resolving gel, the chloride ion always will move, now followed by glycine anion, followed by our protein. So this is the change that takes place in the resolving gel. Now the chloride anion and the glycine anions will move faster in the gel, leaving behind the protein molecules. Now the protein molecule will separate based on the molecular mass. Lower the molecular mass, higher the movement. Higher the molecular mass, slower the movement. So accordingly, based on the molecular mass, the protein separation will occur in the resolving gel. Of course, we use bromophenol dye, and that dye is going to be called as a tracking dye. And that is not going to play any major role in the electrophoretic separation of protein. That is only to know whether our protein has been separated fully or not. And other than that, bromophenol blue dye has nothing to do with this electrophoretic separation of proteins. And once the proteins are separated, and we will once the separation is over, the gel has to be stained with the standard dyes. Usually, most commonly used dyeing method is Kumasi Brilliant Blue R250 staining. And we have the other sensitive staining methods like silver nitrate staining. And whatever the case, ultimately what we are going to see, all the proteins. So here are the proteins. And I must also tell you the purposes of electrophoretic techniques, native page and SDS page. Native page is primarily used for analyzing the isoforms of proteins, isoforms of enzymes. And SDS page is specifically used for the molecular mass determination of a particular protein. And if you want to compare the protein profile between two different conditions, two different organisms, then one has to use SDS page. For example, if you want to compare the protein profile between normal cell and cancer cell, SDS page can be used. But at the same time, if you want to see the isoforms of an enzyme, isoform of a protein, and the SDS page will not be ideal. So therefore, Under this circumstance, native page has to be performed. And one more information, and the molecular mass cannot be determined with the native page. If you want to determine the molecular mass, then the best method to determine the molecular mass is SDS page. So these are the two purposes for which SDS page is performed. The first purpose is to compare the protein profile between two different conditions, two different organisms, and two different treatments. For example, if you want to grow bacteria at lower and higher temperature, if you will grow one bacterium at 30 degrees centigrade, another bacterium with 60 degrees centigrade, and you want to see the protein expressed by the bacterium at two different conditions. So under these circumstances, you'll have to use SDS page. Similarly, if you want to compare the protein profile between the normal cell and the cancer cell, so what you have to do, you have to extract the protein from the normal cell and as well as the cancer cell and run the SDS page and see the protein profile. And the bacterium which was grown at an elevated temperature will express some newer proteins that will not be available in the normally grown bacteria. So these are all the differences that we want to see on SDS page. And so the next technique, the next development that occurred in the electrophoretic technique is called isoelectric focusing. In the previous cases, I described about two different techniques, native page and the SDS page. The separation of protein in the native page is based on the net charge available on the protein molecule. Each and every protein will have a charge on the protein surface. But as far as the native page is concerned, the separation is purely based on the net charge available on the protein molecule. But if you come to the SDS page, where the separation is primarily based on the molecular mass, because all the proteins will be converted into negatively charged protein after the treatment of SDS, and therefore the only option, the only factor which will separate the protein is the molecular mass. And lower the molecular mass, higher the moment, and higher the molecular mass is lower the moment. So these are all the principle behind the native page and the SDS page protein separation. But the third technique now I'm going to explain is the isoelectric focusing. And here, and the factor which will influence the protein molecule to separate is the pH. Each and every protein has its isoelectric pH. Isoelectric pH is nothing but the pH at which a protein molecule will become stationary. If you want to run a protein mixture in a isoelectric focusing gel, then one particular protein will become immobile at a particular pH. For example, protein number 1 will become stationary at pH 4. And here is the electric field. And you load your protein sample, mixture of protein sample, and the isoelectric focusing gel is prepared with the pH gradient. Whereas in the previous two cases, native page and the SDS page, and these two gel systems do not have the pH gradients. Whereas the isoelectric focusing gel will have the pH gradients. So the leftmost end will be pH 5 and the rightmost end will be pH 8 or more than 8 also. And here is a mixture of protein, and the mixture will have a variety of proteins, hundreds of proteins, thousands of proteins will be there. And some of the protein will have isoelectric pH 2. If your protein has isoelectric pH 2, and that protein will become stationary at pH 2. And if the isoelectric pH of another protein is 5, and that protein will move here and will become stationary and will not move any further. And like that, if another protein is pH 7, isoelectric pH of one protein is a 7, and that protein will move here and will become stationary. Likewise, the mixture of protein will move towards the pH gradients and will stay at a specific pH. And between pH 2 and 3, there will be 2.21, 2.3, 2.4, 2.5, 2.6, up to 2.9. And proteins, there are a variety of protein will separate based on the isoelectric pH of the protein. Okay, so here the protein separation is purely based on the isoelectric pH of the protein. And now the isoelectric focusing gel, when it was discovered in a larger format, like the SDS page and the native page size. But so much of developments have occurred, and now the isoelectric focusing gel is exactly the same thickness that I have drawn here. So this is the thickness of the isoelectric focusing gel. Whether it is in a larger format or in a thin format, the isoelectric gel has to be incorporated with ampholytes. Ampholytes are small molecular weight pH gradients. And we can prepare the same, we can use the same protocol which is used for preparation of native page and SDS page. The same way, we can also prepare the isoelectric focusing gel, but only variation here is we are going to add ampholytes. And the ampholytes, we have different ampholytes molecule. One ampholite molecule will be specific for pH 2. Another ampholite molecule will be specific for pH 6. And another ampholite molecule will be pH 7. So ampholytes are a mixture of small molecules. Ampholytes is nothing but these are all small molecules. Yeah, mixture of small molecules which will have specific pH. So, and after the polymerization of the gel has to be incorporated with the ampholite molecule. And after the incorporation, the gel has to be connected to the positive and the negative electrode, and a very high amount of electricity has to be applied. For example, 1500 volt electricity has to be applied. Under this high voltage, the ampholytes molecule will separate and will form pH gradients, starting from pH 1. So here is the pH 1 and pH up to 10. Okay, and so after the pH formation, then you'll have to load the sample, protein sample, and the protein sample will move towards the ampholytes, the pH gradients. And if your isoelectric pH of your protein is 2, then it will stay at pH 2, and other proteins will move towards the other pH and will become stationary in their respective positions. Okay, so this is the basics between isoelectric focusing, and in the isoelectric focusing, the protein separation is based on isoelectric pH of the protein. Now comes another variation called two-dimensional electrophoresis. So this is actually the newer technique, two-dimensional electrophoresis. And the two-dimensional electrophoresis will have separation of proteins on 2 dimensions. One dimension of protein separation, the first dimension of protein separation will be always on isoelectric focusing gel. Remember, the size, the actual size of the isoelectric focusing gel is almost 1 millimeter in size. And you may ask a question. If the size of the gel is 1 millimeter, where exactly we will load our samples? We don't have to load our sample, but what you have to do, you have to take a tray, a rectangular shaped tray, in which you put your isoelectric focusing gel, which will be about the same size of a single string of a noodle. And then put that isoelectric focusing gel, and then it has to be immersed in a sample containing proteins. And the protein sample, the gel now has been immersed into the sample, and the protein will absorb, and the protein will bind, and the protein will be impregnated into the isoelectric focusing gel. And after the impregnation of the isoelectric focusing gel, and it has to be connected to the electrodes, and the power supply has to be provided to the electrodes. And now the protein separation will take place based on the isoelectric pH. And after the separation of the protein in the isoelectric focusing gel, the gel has to be removed from, there is a specific apparatus similar to the apparatus we had for native and SDS page. We also have a specific apparatus for isoelectric focusing, and the gel has to be connected to the apparatus, and the protein will be separated. Now the protein will separate based on the isoelectric pH of individual proteins. And after the protein separation, the gel has to be removed, and you'll have to prepare SDS page. And after the preparation of SDS page, the isoelectric focusing gel has to be placed on top of the SDS page. And the isoelectric focusing gel has to be aligned and molded on top of the SDS page. And this gel will be connected to the upper tank and lower tank in an electrophoretic apparatus. So the upper tank will be connected to negative electrodes, and the lower tank will be connected to the positive electrode. Now the protein separation will take place. Now the separation is in the SDS gel. Can anyone tell us how the protein separation will take place in an SDS page? And the protein separation in the second dimension, that is on SDS page, will be based on molecular mass. Remember whatever I said before when I was talking about the SDS page. In the SDS page, the protein separation will take place based on the molecular mass. But now we are combining two different separations. One separation which was done based on the isoelectric pH of the protein. And now we are combining another separation, SDS page, where the protein separation will take place based on the molecular mass. So we are combining these two techniques together, and the combined technique is now called two-dimensional electrophoresis. And the advantage of two-dimensional electrophoresis is, for example, here are a single protein. In the single dimension of separation in the SDS page, you get only a single band. But when you go for the two-dimensional electrophoresis, the single protein now split into several different fragments, and now a single protein now has yielded more than 10 different proteins. all the protein which come in the straight line in the second dimension of gel or come from a single protein. So this is the major advantage of the two-dimensional electrophoresis. Similarly, the protein here, this is the protein which was a single band in the single dimension of electrophoresis. But after we did the two-dimensional electrophoresis, the single protein has now split into several different proteins. And what we can do, and each of the protein spots can be removed specifically. And there is a machine called spot picker, and which has a software, and if you provide all the details, which band you want to analyze, accordingly the machine, the spot picker, will pick one particular protein band, and this protein band alone can be removed from the gel, and you can go for any kind of analysis. You can go for amino acid analysis, and you can go for whether this particular protein has any activity, biological activity like antibacterial activity, antifungal activity, anti-cancer activity, or anti-inflammatory activity, any other activity you can evaluate with the protein. And before the two-dimensional electrophoresis, and the single protein that you observe is not a single protein, it's made up of several different proteins. And because the molecular mass of 10 different proteins, if the molecular mass of 10 different proteins are similar, then all of them will resolve at the same place. But since we have introduced a technique called isoelectric focusing, where the separation is based on the isoelectric pH, the first dimension of separation will take place based on the isoelectric pH, and the second dimension of separation will take place based on the molecular mass. And ultimately, what we are going to get, thousands of proteins, separation of thousands of proteins, which cannot be achieved with normal native page or SDS page. And I also would like to introduce another technique which has come up lately. And this electrophoretic technique is called the differential electrophoresis. Differential electrophoresis involves a two-dimensional electrophoresis. You know now, at least now you know, two-dimensional electrophoresis is performed with the two different electrophoretic techniques, combining two different electrophoretic techniques. The first technique is isoelectric focusing, and the second technique is SDS page. And now, in a single gel, I want to see the proteins expressed by a normal cell and the protein expressed by a cancer cell. We have normal cell here and we have a cancer cell, but I want to see what specific proteins which are present in the normal cell and what specific proteins are there in the cancer cell. So what I normally will do, I will extract a protein from the normal cell, also from the cancer cell. But after the extraction of the protein extracted from the normal cell will be combined with a dye, a fluorescent dye. Normally, there are several different dyes, fluorescent dye molecules are available. The most commonly used dyes are Psi3. Remember, this is the normal cell and we extracted protein from the normal cell, and after extraction, all the proteins are conjugated with the Psi3 fluorescent dye molecule. So all the proteins will fluoresce green, while the proteins isolated from the cancer cell will be conjugated with another fluorescent dye molecule called Psi5, and this dye will fluoresce red in color. And after that, what I'm going to do, so here is the vial which has a protein isolated from the normal cell, and the proteins has already been conjugated with Cy3 fluorescent dye molecule. And here is the vial which has the protein molecule isolated from the cancer cell, and they have been already conjugated with Cy5 dye, which will fluoresce red in color. So now I'm going to take a 50 microliter from the protein isolated from the normal cell, and I will also take a 50 microliter of protein isolated from the cancer cell, and I will mix them together. The total volume will become 100 microliter. And this entire 100 microliter of sample will be run on isoelectric focusing gel. Please remember, the separation of proteins in a isoelectric focusing gel is purely based on the isoelectric pH of the protein. And after the isoelectric focusing separation is over, the second dimension of separation will be performed on SDS page. And the two-dimensional electrophoresis, assume that you have prepared the protein from normal cell and cancer cell, mixed them, and electrophoresed on isoelectric focusing gel, and also electrophoresed on SDS page. Now the two-dimensional electrophoresis is performed. But what is the difference between the previous two-dimensional electrophoresis and the two-dimensional electrophoresis now I am describing? The only difference is the proteins isolated from the normal cell has been conjugated with Cy3 fluorescent dye, and the proteins isolated from the cancer cell has now been conjugated with the Cy5 fluorescent dye molecule. Okay, so now the isoelectric focusing separation is over, SDS page separation is over, and the gel is now ready for viewing under a scanner. So the Cy3 fluorescent dye molecule will absorb at a specific nanometer wavelength. Similarly, the Cy5 fluorescent dye molecule will fluoresce at a specific wavelength. So what we do, we select the nanometer that is optimum for the green fluorescence, so that is Cy3 dye. And we get an image like this. So at a particular wavelength, for example, if you use a wavelength 490, this is only an example, not exactly. And this wavelength will be optimum for excitation of the fluorescent molecule of Cy3. And we use a 600 nanometer to excite the another fluorescent molecule, which is Cy5, and we capture the image. All the Cy3 fluorescent molecule will fluoresce at 600 nanometer, and we get an image. And at 490 nanometer, only the green fluorescent protein will fluoresce. And the images is taken separately with the 490 nanometer and the 600 nanometer. And these two images, with the help of a software, will be merged. And after merging, you will see only red colored proteins. And whenever you see all red colored proteins, these are all the proteins. All these red colored proteins are exclusively expressed in cancer cells. And if you see green colored spots like this, green, green, okay, green, And all these proteins are exclusively expressed in the normal cell. And if you happen to see orange colored bands, those orange colored bands are expressed both in normal cell and cancer cells. So the green and red, if green and red is mixed together, the final color will be orange. Wherever you see orange, that indicates that that is the protein which are expressed both in the normal cell and the cancer cells. Wherever you see green colored protein, and these green colored proteins are very, very specifically expressed in the normal cells. Wherever you see red colored proteins, and these are the proteins which are exclusively expressed in the cancer cells. So if you want to see the functions of the protein which are exclusively expressed in the cancer cell or in the normal cell, and you can remove the protein separately and remove the protein expressed in the normal cell separately, and if you want to see the functions, and you are very well in a position to see the structure and the functions of the proteins. And so this is the latest development in the analysis of proteins, proteomics. It is called DIGE, differential electrophoresis. And organizers, please tell me if I can take another 5 to 10 minutes, or you are running short of time. I don't hear anybody.
Sir?
If I can go further for 5 to 10 minutes, is it okay, or should I wrap?
Sir, 5 minutes is okay, sir.
Okay, fine.
Thank you, sir.
Okay. And the next technique in the proteomic analysis is the Western blot analysis. And many of you may be knowing what is Western blot analysis. If you want to detect the presence of a protein in an organism, Western blot analysis is performed. And previously, only a single protein will be detected with the specific antibody combined with the secondary antibody. So here is the outline of what happens in a Western blot analysis. So here is the antigen molecule, and the antigen molecule is recognized by the primary antibody. And the primary antibody now recognizes the secondary antibody. The secondary antibody is conjugated with an enzyme. And depending upon whether the enzyme activity appears or not, you will say the presence or absence of protein. If the color appears, you will say that the presence, that particular protein is available. If no color development occurs, you will say that that particular protein is absent. Now, similar to DIGE, in the Western blot analysis, previously only a single protein can be detected with the specific antibodies. But now, you can combine many different antibodies in a single gel. So the protein isolated, you can test this is organism 1, organism 2, and organism 3. And if you want to see the presence of a particular protein, and the protein isolated from this cell type can be conjugated with the antibody, specific antibody. Similarly, the protein isolated from this second cell type can be interacted with the secondary, another antibody molecule. Similarly, the protein isolated from the third cell can be interacted with the third primary antibody. Not only that, the primary antibodies can be conjugated with the different fluorescent molecules. Previously, and this technique was not available. But now, after the discovery of several fluorescent molecules, and here is the primary antibody 1. Primary antibody 1 can be conjugated with the Psi3 dye. Second, the primary antibody specific for the second cell can be conjugated with the Psi5. And the primary antibody, which is specific for the third cell, can be conjugated with Cy7, which will fluoresce blue in color. So, in this particular gel, the detection of two different protein at a single time can be done. So, this is the fluorescent molecule Cy5 incorporated protein molecule, and here is the Cy3 incorporated protein molecule. In a single gel, you can detect more than two different protein, presence of two different protein at the same time. Only thing is, you will have to use different primary antibodies specific for the protein of interest. Okay, so this is about the Western blot analysis, and now comes the mass spectrometry analysis. Of course, I do not have enough time to explain the working principle of mass spectrometry, but this is mass spectrometry is basically used for detection of several post-translationally modified proteins. And there are several post-translational modifications in a cell. In eukaryotic cell, there are several post-translational modifications. And these post-translational modifications can be broadly grouped into reversible post-translational modifications and irreversible post-translational modifications. So under the reversible post-translational modifications, we have hydroxylation. Addition of hydroxyl group to a target protein is hydroxylation. Addition of phosphate group to a target protein is called phosphorylation. Addition of acetate group to a phosphate to the target protein is acetylation. And then addition of methyl group to a target protein is methylation. And in addition to hydroxylation, phosphorylation, acetylation, methylation, we have other post-translational modifications of the protein. AMPylation, ADP ribosylation, glycosylation, isoprenylation, and the UBL protein conjugation, ubiquitination, deamidation, elimination, proteolysis. Okay, these many post-translational modifications of proteins are there. But how are we going to detect all of them? They normally use the SDS page. Even the two-dimensional electrophoresis will not detect the post-translationally modified proteins. So in such circumstances, we use the mass spectrometry. And this is the latest technique available in the analysis of proteins. So in a given organism, for example, in a small piece of plant tissue or in an animal tissue, how many proteins are phosphorylated? How many proteins are methylated? How many proteins are acetylated? And all these post-translational modifications can be detected with the mass spectrometry instrument. And this is the latest development in the proteomics. And all the post-translationally modified proteins can be analyzed with the mass spectrometry. So these are all the several advancements that have taken place in the analysis of proteomics. But in fact, I have not included everything that involves in the proteomic analysis. I have selected only few of them. There are other techniques also involved, but that will be too much for the participants. So I restricted myself with only few of the techniques used for the proteomic analysis. And with this brief note, I thank you all for your patient listening. And if at all there are questions or clarifications you have, you can very well ask me now or maybe later also. And before I could conclude my lecture, I would like to thank one more time the organizers, the faculty members of Department of Botany, Department of Zoology, and the administrative officers of the Avinashilingam University for giving me this wonderful opportunity to speak to participants from several regions of our country. And thank you all for your patient listening. Thank you.
Thank you very much, sir. Your session was actually mind-blowing. Thank you, sir.
Thank you.
Thank you, sir. Now the session is open for queries and clarifications. If any of the participants have any queries, you can actually post them in the chat box or you can unmute and ask, sir. Do we have any queries from the audience side? Okay then. Thank you, sir. Your session about proteomic analysis was very informative and we learned a lot starting from the very basics of proteomics. We're blessed to have you, sir. Thank you so much.
Thank you very much.
Now I request Mrs. A. Uma Devi, Head and in charge of Department of Physician Assistant, to propose the vote of thanks.
Very good evening, one and all present here. Gratitude is the inward feelings of kindness received. Thankfulness is the natural impulse to express the feeling. Thanksgiving is the following of that impulses. So first of all, we thank God Almighty for giving us strength and support for made this online training program to reach great heights. The path of success in the society has to be achieved through refined education and strenuous labor. I feel that the path of success is through this online training program. First of all, I would like to thank Dr. K. Manimoli, Professor, Department of Resource Management, and the Project Coordinator of UGC, STRIDE, Component 1, for giving us this opportunity to organize this event. Thank you, ma'am, for giving us such an opportunity. On behalf of School of Bioscience, the Avinashilingam Institute, our sincere gratitude to Dr. B. Palani, Professor, Center for Advanced Studies in Botany, University of Madras, for his immense presence and disclosing their precious lecture on recent advancement in proteomic analysis. It was really informative, full of knowledge and interesting things. So thank you for your informative session, sir. Thank you, sir.
Thank you, madam. Thank you very much.
I would like to express my special gratitude to all the esteemed resource persons who have accepted our invitation for these 6 days. They enlighten all of us with their informative session. Really, all the participants are benefited from these 6 days online session. Thank you all delegates. I wholeheartedly thank all the higher authorities of our institution for supporting and encouraging us in all our academic endeavors. I express my sincere thanks to Dr. A. Vijayalakshmi, Dean, School of Bioscience, Professor and Head, the Department of Botany, for your marvelous guidance and support to complete this online training program in a grand manner. Thank you, ma'am. I am grateful to thank our conveners, Dr. Anita Subhas, Professor and Head, the Department of Biochemistry, Biotechnology and Bioinformatics, and Dr. K. Manimekalai, Professor and Head, Department of Zoology, for always encouraging and guiding us in all the proposed events in an organized manner. I thank both of them for making this event a great success. My sincere thanks to all the organizing secretaries, Dr. G. Mageshwari and Dr. B. Kartiga, Assistant Professor, Department of Zoology, and M.K. Nisha, Dr. V. Gayatri, Assistant Professors, Department of Botany, Dr. S.T. Yamuna and Dr. C.C.S. Vasundhara, Assistant Professor, Department of Biochemistry, Biotechnology and Bioinformatics, Mrs. Ashwini G., Assistant Professor, Department of Physician Assistant, for organizing this online training program in a smooth and pleasant way. Thank you all for your constant support, teachers. I take immense pleasure to thank all the faculty, participants, scholars and students from various parts of India and our institutions for participating in the training program and making it as a successful event. Thank you all participants. I also wish to express my gratitude to all the dedicated and hardworking faculty members from the Department of Botany, Zoology, Biochemistry, Biotechnology and Bioinformatics and Physician Assistant for the constant and untiring support all the time. Last but not the least, I thank our Dr. Meena and Mr. Ramesh, Computer Department for the technical support and services in live streaming. So once again, I thank everyone for your presence and support. Thank you all. Thank you, sir. Thank you so much.
Thank you. Can we log out? Can I log out?
Yes, sir. Yes, sir. Thank you.
Yes, sir.
Thank you so much, sir. Palni sir, thank you, thank you.
Thank you very much, madam.
Yes.
With pleasure.
Thank you all. The link for the feedback form has been posted in the comment box. Participants can find their link in the chat box here. Thank you.